Riuscite a vedermi e sentirmi?
Sì, Alice, ti vediamo e ti sentiamo.
Ok, grazie.
In un minuto cominciamo la sessione. So welcome back for this afternoon session dedicated to pharmaceutical technology and drug delivery. We start with the second keynote of the day, and in particular, we are hosting Professor Dimitris Lamplou, which is a reader in pharmaceutical engineering and program director of the Master of Science in Industrial Pharmaceutic at the School of Pharmacy in Queen's University, Belfast. He's author of over 100 peer-reviewed publications and over 250 conference abstracts, and he has over 100 oral invited talks in institutions and conferences all around the world. Furthermore, he has also secured during his career a funding in excess of £2 million. His research and academic leadership have been recognized with a range of awards and membership to society and editorial board. For example, he is right now chair of the United Kingdom Environmental Control and Release Society and also chair of the Emerging Technology Focus Group that is supported by the Academy of Pharmaceutical Science. His research focuses on emerging micro and nanofabrication technique to produce drug delivery system. And for instance, he's an expert in 3D printing, bioprinting, electrospinning, and microfluidics. The general aim of his work is improving the delivery, the delivery of all drug through drug delivery system, at the same time supporting the development of new personalized medical device. But let's hear and learn more directly from him through his talk titled Transforming Treatment Through Emerging Drug Delivery Technologies. Dimitris, if you're online and ready, okay, we'll see your screen.
Hopefully you can see me, you can hear me also, Gabriella.
Yes, we can hear you very well and see your screen. So the floor, or actually the screen, is all yours.
Thank you. Thanks so much, Gabriella, and thanks the organizers for the amazing event for the last days. I've enjoyed a lot the presentations and also nice posters from young scientists. It's good to see that. So as Gabriella mentioned, my name is Dimitri. I'm based at Queen's University of Belfast. Today I'm going to talk to you about how we're using emerging healthcare technologies for drug delivery applications for pharmaceutical technologies, but the new technology, if we want to call them new. If you're not familiar about Belfast, we are in Northern Ireland. 1 hour flight from London or a couple of hours driving from Dublin. There are some amazing views just half an hour from where I am now in the city center of Belfast, based in the School of Pharmacy. And this is one of our historical buildings here, also the Queen's University of Belfast, that is the 9th oldest university in the United Kingdom. So what about emerging technologies? Emerging technologies, they are transforming the pharma world. And emerging technologies like artificial intelligence for drug delivery research development, bioinformatics, can be body sensors and silico trials, but also technologies like 3D printing, bioprinting, microfluidics, personalized and precision medicines, and continuous manufacturing has been always there as an emerging technology. The ones in green is the ones that we do here in my lab. My lab is divided in 3, if you want, research areas, which the first one is electrospinning and melt solution. So we do a lot of electrospinning for drug delivery, wound healing applications, and for cell scaffold applications also. And we have a variety of systems in our lab. Now, the next one that I will spend more time here is about microfluidics and lab on a chip. So we using microfluidics for the manufacturing of polymer and lipid-based nanoparticles, and we compare those with traditional, if we want to call it traditional, formulation methods. We also, because we have over 20 3D printers in our facility, we're using also the 3D printers to prepare different chips for using it either as a lab on a chip or using it for our nanoformulation applications using microfluidics. Now, I'm sure most of you're familiar with this. I saw a few also posters around that. Microfluidics is precise control and manipulation of very small fluid flows. And this translates to small amounts of reagents used. Simpler is faster. It can be also in continuous and can be also scale up. Of course, we have to take case by case. We can't say in generic that it can be scale up for everything, for example. Okay, this has to be case by case always. The microfluidic market is not totally new. It's there for over 20 years probably, but the last years has increased a lot the interest, especially the last 4 to 5 years. There's a lot of interest in drug delivery. Mainly the microfluidics research or also development with a lot of different applications is in drug discovery, drug delivery, in vitro diagnostics, a lot also about COVID applications with microfluidics as lab on a chip also, but also as in vitro diagnostics for the last couple of years research. Now, a few years back, a few scientists, including myself, we came together to explain what's the issue of transparency and reproducibility in nanomedicine, and we wrote an article in Nature Nano. Inside that article, we explain about issues like part sizing is a very important issue, but manufacturing also, like how you make sure that you always have the same monodispersity and release and stability of your formulations. And of course, microfluidics, it is one of those methods that you could use for such applications. Because you can have more tunable features, that means you're controlling the diameter, the structure, and the surface also. You can have a good robustness, low batch-to-batch variations, control release, precise control release, and good versatility between micro, nano, and also what molecules would like to encapsulate. And I will provide you a few examples with different molecules, including biologics, because this is very important now, and this is a lot of interest we're getting from our collaborators in biopharma, especially after the vaccines for COVID, for mRNA, for example. Now, microfluidics, you can have different channels, you can have different flows and this. The most common one has been used for the production of liposomes, say, and PLGA, is by using 2 inputs and 1 output. Now, your channels can be completely different inside. This is just one typical one has been used a lot in the literature. As I mentioned, we're using a lot of different chips and designs because we can make that using 3D printing, and I will show you in a moment. We have 6 different platforms in our lab, including our own in-house setup that we prepared from scratch. But we have a lot of good collaboration with different industries and having different systems in order to make sure that it will be good for our applications, but also at the same time that we can look about stability, we can look also about scale up, and also continuous manufacturing and everything to be also in a clean room environment, also to be easy to transparent that. Now, we're all familiar about COVID-19 and lipid nanoparticles. There's been a lot of research in the last couple of years, especially lipid nanoparticles. It is one of the main components of the Pfizer vaccine. Of course, we can use microfluidics for the production of nanocarriers like liposomes, and we can use it with encapsulation of sRNA, for example, or other biologics on that. And I have done that also in our labs. Now, let me show you a few case studies from our lab. Then you can see here two particle sizing results. One is with a traditional method, and the other one is with microfluidics. Hopefully, you can spot on which one is which. Because as I mentioned already, with the microfluidics, you can have more narrow down dispersity, monodispersed particles, and you can control the size. So it's the one is my mouse pointing here. Now, by control, you can control the flow rate and also, flow rate, how quick we get inside those channels, but the flow ratios also, the flow ratios between the two channels or the three channels, the inputs you have in it. It depends on your application. And as I said, this will be case by case applications, of course. And it depends on the flow rate and the flow ratio with exactly the same formulation, exactly the same drug. We can control the particle sizing. Then, as soon as we know the particle sizing that is the best for us, we can start controlling the release. So we can control the release by changing some of those formulation parameters. And we are building a database by using, say, specific lipids, phospholipids, and specific also drugs. What flow rates you change in a flow rate or parameter, other parameters, temperature, for example, solvent, that you'll be able to have a specific release or a specific sizing or other applications. So we do that a lot in making the databases for that. Now, another application, another case study is we're using, we have a very strong collaboration with University of Pavia, and I'm there also, I'm going tomorrow as a visiting professor, and we're using nanoparticles for biomedical imaging. So contrast media has been used for imaging. Now, the contrast media, they're slightly toxic, and one of the issues is contrast agent administration, which is the third most common cause of hospital-associated renal failure. So now you want the opposite. Now you want high encapsulation efficacy inside your nanocarrier, but you don't want any release because you don't want to be toxic in your body. So we have reported that for first time. We're using liposomes. We have in the release very, very tiny one that wasn't toxic in the cells that we are studying. But we said also, wait a moment here, what about also that small release we had? How about if we put also another agent, another drug inside that if you had this problem, the contrast media releasing, then you have a drug to support you not to get any contrast agent administration on that. And that is, any hospital-associated renal failure with the contrast agent administration. And we put Plga nanoparticles, and so we produce Plga nanoparticles using microfluidics with the University of Pavia, and then we managed to have two molecules in the same time. You can find more information in our recent paper in the International Journal of Pharmaceutics just a couple of months ago. Now, we want to study to see what's happening, of course, when we do those particles inside the cells, yeah, because you have in the particles. So this is a cell structure under atomic force microscopy, a bio-AFM. Now, this is under the cell. We're changing the height. This is exactly the same as here, but we change the height because now as soon as we put the nanoparticles, the height will change, and we would like to see what's happening. And you can see here now the nanoliposomes start getting inside the cells. And that's what we would like to see also for some cancer applications, especially when we're targeting the cancer with specific particles and producing with microfluidics, for example. Now, another case that we have in collaboration with Portugal is about target drug-loaded nanoparticles for neurodegenerative diseases. And we all know the issues about delivering particles for BBB. And so we wanted to deliver a specific drug molecule and for specific sizing also and for specific time, which I cannot say today all of those information, but what I can say is we play with different parameters as I mentioned. So, and we're able to producing, of course, the particles that is over 200 nanometers, as you can see here, they're not good, but we are able to produce particles down to 50 nanometers, for example, with high load in encapsulation efficacy that they can be used also for BBB applications. So they will be stable, those particles, and they can release the drug through the BBB. Now I'll say, okay, yes, you're telling us about drugs, molecules, but what about biologics, which is, this is the question now in 2021 with all the vaccines. Yes, we have done a lot of biologics also. I'm not able to show you everything because this is a collaboration with biopharma companies. So, for example, here is a collaboration with one of the biological companies in UK, and the results I can show you here is about BSA loaded liposomes, and we managed by using different lipids and also different concentrations of the BSA, because it depends on concentration you would like to use and the release profiles you would like, and also if you want to be stable in room temperature or in the fridge or in your body 37 degrees. So we've done all those studies, and we have managed to prepare also particles that can be stable also in room temperature, for example. So you can have it in outside for different applications from the BSA. Now, I mentioned about 3D printed chips, and this is what we do a lot. So we producing the different chips, and that I will move to the 3D printing, which is the next part of this presentation. So we producing the microfluidic chips, and we have a different designs. This is just a simple design to explain it, which we published in the National Journal of Molecular Sciences a month ago also, and in collaboration with the University of Pavia. You can see here exactly the same chip, exactly the same flow rates, exactly the same particles, liposomes, but you're getting completely different sizes, and also by changing the flow rates, because if you want to produce it faster, say to scale up also in faster production. Now, what's the difference between those two? Why? Because we change slightly the channels inside. So it doesn't matter now if we increase in the flow rates, we have increased it also higher, we can get exactly the same particle size always for this formulation by changing the parameters inside. And that's for us easy because we have the printers, we have the capabilities of designing this, and in 1 hour we can get out these microfluidic chips and test them after our printing. And that's something we do. We also using different bioprinters also. This is using a bioprinter. For example, this is a photo I took with my phone a few days ago. And this is by using bioprinters, and we can use biological materials for lab on a chip applications also at the same time. Now, for the last 10 minutes, let's move to the additive manufacturing or 3D printing. For example, we're doing a lot of different 3D printing. So we manufacture different drug delivery systems and medical devices, and we're also producing our own filaments and our own also bioinks for our bioprinters we have in. We are very strong on this. We are on the top 0.1% in the whole world of printing based to statistics for PubMed on that. So 3D printing is due to its small size and affordable prices are convenience and can be used in any environment, and avoiding with supply chain issues that we saw a lot last year due to the pandemic. Now, there are several 3D print technologies in existence. I will not go through all of them because of the time, but in our lab, we have every existing technology, 3D printing technology. We have over 20 printers covering every available type of printer you can imagine currently. And you'll say, why you need this? Because every material is different. You cannot use the same printer for different materials. Either it's a temperature sensitive materials or decantation or anything. So if there is a powders or if you don't want to use a solvents or it's a liquids or on that. So we're using the printer based on the application. So we look at first the application, we understand first what we have to do, and then we're choosing the printers at the end of this manufacturing method. Let's go case by case. I'm sure all of you will love to have chocolate as your formulation. Yeah. How many times your grandpa or your mom or you said to your children, well, if you take your medication, I will buy you an ice cream or a chocolate. Of course, the idea is not to have Nutella intravenous. I'm sure a lot of us would love to have Nutella intravenous, but that's not the idea. There are already formulations out there that are using vitamins in combination with chocolates, so for children. And what we do is we're using in collaboration with Portugal, we're using formulations to make it 3D printed with chocolate for pediatric formulations, for example, to use in hospitals for safety currently, because you don't want to have, including vitamins, you don't want to have chocolate vitamins in your house and the kids taking too much vitamin, but also especially for drug molecules. Now, why pharma is interested in 3D printing? Because of the flexible dose control systems, the complex formulations you can have with multiple APIs inside, and provides a solution to the early phase formulation challenges. And hopefully we can do it for personalizing medicine and produce drugs, produce formulations in pharmacies and hospitals in the future, of course. There are a lot of advantages of using polymeric dosage forms, for example, for long-acting products and also for controlled drug delivery of APIs. So you can improve the pharmacokinetic profiles, and that's what we do. We're using a lot of different long-acting implants also produced by 3D printing applications. There are a lot of drug molecules under investigation. I will not go through all of them, but just a few that there are studies with polymer extrusion, which means we can use it also to produce filaments, and we can use it for different implants or drugs, tablets, for example. CQ in combination with oseltamide has been used for some of the studies. However, that can cause to some patients dose-dependent increase in heart abnormality. So you need to be very careful. So you need to control the dosage from patient to patient. So you can, but if you can do that with the printer very quick, that it will save you time and also lives. Now, there's another one, UK clinical trials under the code name Recovery that shows that low doses of dexamethasone in hospitalized patients with COVID-19 reduce mortality by the 1/3 among patients in serious conditions. Now, filaments can be prepared, so we can prepare filaments using those drug molecules. So using a polymer extrusion to preparing on those, and then formulations can be printed in accurate dosage depending from one patient to the other when they're hospitalized, for example. So you can manufacture, as I mentioned, you can use your filament extruder, and then you're producing your filament or your pasta, as I make it, as I call it. So you have the pasta maker, you have your pasta, and then you're printing. It depends on the designs, and you can have any designs you like. And it depends on the strains you have inside. You can have a different also concentration, and it looks like exactly the same size, same shape as in normal tablets. And of course, you can have a different design for pediatric applications if you like. This is just Q with Queens, for example, that we're using. Now, another case, they were one of the first groups together with, in collaboration with London, and we reported back in 2018 3D printed microneedles. And there's a lot of papers now, of course, after that were the first ones reported. And this is one of our first applications of using insulin. And you can see here, for example, we use different designs, but also this is a micro CT because we wanted to see what's happening when we're having the different polymers there, but also the insulin. The other application was that when we did the animal studies, we saw that when we're using the microneedles, which is the blue line, in comparison to the injections, which is the green line here, we're after some time, of course, with IV, the glucose going up again, when with the microneedles, they will continue stay down. Now we're going to the next level of that. We're preparing microneedles that we're using hollow microneedles that it can be one size hollow microneedles, on the other side it can be the solid with your drug, and we can control it and monitor it like lab on a chip microneedle based systems with MEMS devices, for example. So you can control your glucose in the same time, and you can release the drug that is interesting in that time for each individual. Of course, there's a lot of research, a lot of collaborations going on at the moment, and hopefully we'll have some good results in next time. And another application before I just conclude. So it's a 3D printed vagina message. It's a big issue across the whole world. And it was over the BBC News also a couple of years ago. And last year, FDA had to change the regulations about vagina messes had been used because there was barbaric treatment for women, for having pain, blood also, and a lot of other applications. So what we did, so we have done a lot of research in this. We have, I think, a lot of papers on that by using different designs, different drugs, different polymers also, because you have to find the best flexibility, but at the same time to support the organs, but do not hurt the patient, of course. So we have done a lot of studies I mentioned using different materials and different polymers. You can go in some of our papers on that. And we also now moving to do some animal studies to understand a bit more of those applications, and if we can pattern having new products in the market around that area. As I mentioned, we do a lot of different 3D printing because we have a lot of different printers also here. So this is some examples of vascular grafts we do for cardiovascular applications. For hemodialysis, catheters also, different applications for catheters. Biodegradable also drug release substance and drug release systems that you can put it in your body, either for cancer applications or for other applications also. Vagina rings, wound healing applications with bioprinting, but also we support a lot during the pandemic. From the first day, my team was here non-stop, using all of our printers to produce different items for NHS and National Health Centre in UK, and providing our knowledge nationally and internationally on that. And I think I'm on time on this, and just to conclude, so using new technologies, drug system devices can be produced, and new technology like microfluidics and also 3D printing and bioprinting. Microfluidic is a rapid one-step method for the production of small homogeneous for lipid and also Plga nanoparticles, and there are a lot of other studies currently around that. The versatility of microfluidic provides suitable alternative method to other formulation methods also, and as mentioned, additive manufacture 3D printing can be used to manufacture different drug delivery system, medical devices, and implants. I would like to thank, of course, all our sponsors and my team. Without them, I will not be able to present anything today. Postdocs, PhD students, research visitors across the whole world, and especially from Italy, I have 4 or 5 research visitors every year. Currently, I have 3 in my lab. And thank you very much for your attention. Happy to take any questions. Thank you.
So thank you, Dimitris, for this very interesting talk. I have to say, this emerging technique seems very promising. I mean, you showed really nice reproducibility of the system. I mean, a wide open application. Actually, my curiosity is about the limitation. So I would like to hear from you that are the expert, the eventual limitation of 3D printing or microfluidics.
And the limitations, there are a lot, of course, yeah, and that's
we cannot
why
hear you. Maybe you are muted.
Can you hear me now?
Yes, perfect.
Okay, so the limitations, it depends from application to application, of course, Gabriela, and scalability is one big issue for both technologies, for 3D printing and also for the microfluidics. But again, for microfluidics, it will depend of what you would like to scale up and what we meaning about scale up also in the same time, because different people, they call in different things, scalability for microfluidics. Now, 3D printing, there are some printers for applications that they can be scale up, but again, it has to be case by case for the same. I'll say the main issue here will be the scalability and also how we are making sure we making products that with 3D printing especially that will be autoclaved or radiation free also, because you need to make sure that implants, they will not have any infections in your body. Yeah. You don't want to kill yourself, you want to help yourself.
Thank you again, Dimitri. So I see a resident from Claudia.
Hi, Dimitri. Hello. So thank you for your great talk. We have a lot of systems, a lot of technologies are really, really fantastic. I was just, of course, when I saw your presentation, my curiosity gave immediately to the chocolate 3D printing, but doesn't matter. My question is just a curiosity. With the microfluidic, you have a product, you have developed the lipidic nanoparticles, lipidic system. I don't know if you have never tried with the polymeric based nanoparticles.
Yes, the first study I saw you that it was with for the biomedical imaging is the Plga Nanoparticles, and this is with the University of Pavia. But we have also done some Plga with in collaboration with the University of Porto also. So yeah, we're using Plga or lipid-based, I will call them. So liposomes and also solid lipid nanoparticles, because this is solid lipid nanoparticles, I will say, will be the next future after the liposomes now.
Yeah, of course. But do you think, I don't know, I'm not an expert in this field, but it's possible to connect, just a curiosity, different chip, one to each other to have a more sophisticated system? I don't know, to create a Plga nanoparticle, then with another chip, a coating of Plga. I don't know if you ever never tried.
Yeah, we have done that. That's why I'm laughing, Claudia. Yes, I'm not allowed to show results, but yes, we have done that.
Okay,
because
okay.
You need to have us. That's why I mentioned it depends what we're talking about, continuous and scale up. Yeah.
Yeah.
Because you want to prepare nanoparticles, say Plga nanoparticles, and then coat them for stent or anything.
Yeah, of course.
So that's then you, as soon as they're coming out, they're going to the next chip and then to the next one. But in the same time, you need to make sure you have a quality control on that. And we do a lot of PAT from one step to the other. And
anyway, it's a continuous process.
Yes.
You cannot stop the one chip from the other one.
Yeah, we don't stop in it, and that's why we're using PAT technologies like a UV probe, Raman probe, and NIR probes to see what's happening in each step of the process. Unfortunately, as I said, I'm not able to show the data on this. But yes, we do that, and that's why I was laughing with your question, smiling.
Okay, thank you.
I can see a question, Gabriela, yeah?
Yeah, no, we have one more question from Dr. Michaelis Klik. So thank you for sharing your expertise and data. Connected to the question from Gabriela about limitation, what is the resolution you can achieve when printing microfluidic chips, considering that especially for the production of nanoparticle, even small imperfection may have an impact on the quality of produced particle?
I totally agree.
We cannot hear you. No.
Oh.
Okay, sorry. Yeah.
You can hear me, yeah? Okay.
Yes.
Thank you. Oh, yeah. Yeah, and that's a very good question. This is what we did in our recent paper. So We have used 3 different technologies. We used FDM, we used also SLA, and we used DLP. And in order to compare the resolution from one technology to the other to produce exactly the same liposomes. So I will refer in that because of the time in that paper, but more than happy to having also more discussions. But yeah, this is clear, and that's why we did that. And that's a good thing to have all the different technologies in your lab so you can do those studies.
Okay, thank you. Let's see if we have more questions, but I don't see any other hand or text in the chat. So I will thank you again, Dimitris, for accepting our invitation and to present us your work and all the devices that you have in your lab.
Thanks so much, Gabriela, and everyone.
We'll be happy if you can keep following us, and we can move to the next oral communication. We have Dr. Vincenzo Taresco from the University of Nottingham with a talk on polyglycerol adipate, PGA, an enzymatically synthesized functionable polyester as a versatile drug delivery carrier. Vincenzo, we'll see your screen, and when you are ready, the screen is all yours.
Gabriella, can you hear me?
Yes, we can hear you.
Fantastic. Many thanks for the introduction. And let me join Dimitris in saying that you have been amazing in organizing this conference. I also enjoyed the 3 days, and yeah, very well done. Looking forward to coming to Naples next year, hopefully. So, as Gabriella mentioned, I'm going to talk about a bit this polyglycerol adipate. Yeah, it's quite uncommon polymer, so I will try to give you like an overview on why we believe here in Nottingham there might be like a promising polymers to be a replacement of some of the aliphatic polyesters. But anyway, every single time I'm introducing these polymers to any new collaborators, this is the effect that this name produces on people. So how I'm trying to explain and justify the quality of these polymers and to promote these to my collaborators, I will try to do today with you and try to convince you that this is a great polymer, so at least promising polymers. So why we want to replace, at least in part, at least trying to replace amazing materials like the aliphatic polyesters that we have heard so far today about, like PLA, PLGA, polycaprolactone, or any pegylated copolymers. So as highlighted so far today, these are like fantastic materials. They are present in a variety of FDA-approved formulations. They are biodegradable, biocompatible. They can self-assemble nanoparticles, or they can be used in tissue engineering scaffold. So why we want to replace them, or at least find some possible new avenues for new polymers. And this is not just to justify my salary, but because these materials are in effect suffering a variety of crucial limitations. As a polymer chemist, of course, I'm keen on highlighting the problems that are related on the synthetic pathways. And in particular, PEG is coming from a toxic monomer, and there are really drastic reaction conditions to produce that. And then all the biological issues that are related, you know, better than me. Regarding the polyesters part, of course, we know the catalysts that are normally used in the reaction might be toxic, and they should be removed as much as possible. They don't have many chemical functionalities to be used to improve properties like in formulation for interaction with drugs, or to improve the biological properties of these materials. So how we come up with the idea of polyglycerol adipate? Actually, it's not my idea. The polymer was already in literature since early 90s, but it was taken up as a possibility for a drug delivery carrier by Professor Martin Garnett, who had the pleasure to be his last postdoc before he retired, and now he's happily enjoying his retirement in the neighborhood of Derby. And let's say that I've taken up his concept of moving forward some more sustainable, more hopefully clever options for aliphatic polyester. So glycerol. So we start from glycerol. Glycerol is a really simple multifunctional alcohol that is mainly produced as a waste materials from the biodiesel cycle. Just to give you an idea, the production as a waste materials, only as a waste materials from biodiesel, is expected to be around 4,000 or more million liters by 2026. And we are talking about something like over 2,000 Olympic swimming pool put all together of these side products. So we should be able to do something with these. And this is our first part of the molecule. The second part is based on adipic acid, which is one of the most produced diacid in the market, and is used to produce mainly nylon 6,6, but also is used as a food excipient. So it can be metabolized by our body. However, to facilitate the reaction system, we started from a modification of the adipic acid. It's also commercial, it's DVA, divinyl adipate, which is normally used as a cross-linker in tissue engineering, as a biodegradable cross-linker. However, we have a way to get this hydrophilic part and this hydrophobic part in a polymer that can be used in different way in the future. So how we can combine these two and get a polymer rather than like a sludgy gel? So we do that by exploiting the properties of a commercial enzyme. Is a lipase. It's called commercially Novozyme 435. Is a really simple one-step, one-pot reaction. So the enzyme is catalyzing the transesterification between the two molecules, and this is happening mainly in an organic solvent. And what we noticed while we were producing these amphiphilic by default molecule, that by changing the temperature, we can modify deeply the structure of the polymer. So if we work around 60 degrees or above, we end up with a really branched polymers. It's, of course, branched polymers that have like nice properties, but today I want to talk about how we can maintain these free OH along the backbone of the polymers. And let's say this is going to be the key protagonist of this talk. And we can get a free OH for each chain by simply reducing the temperature of the reaction, not changing anything else. However, just as a quick detour before starting talking about the use of this OH, lately in my new group, in particular with the work of a great PhD student, Pippi, we've been trying to enhance or enlarge, broaden the chemistry we can do with these two molecules in some commercial diodes. So just simply changing the stoichiometry of the reaction and playing with a more greener solvent, so just to improve the sustainability of the system, we ended up with a new PGA library of molecules where we managed to add some new functionalities that can improve the self-assembling and the stability of the nanoparticles at the end, giving like more chemical handles for further modification or new responsiveness. However, as I was mentioning before, let's talk about this free OH. So the free OH is giving us hydrophilicity, so it's helping the polymer to self-assemble without the use of any extra surfactant. However, we can also use that to chemically attach a drug or any entities directly on the backbone of the polymers. So in that way, as from the figure, we can like have a really good hold of our chemicals without any leakage that normally is happening when we're encapsulating some small molecules. However, because we can create responsive bonds, if we apply a perfect external stimuli, we can release our cargo, or in some cases degrade completely the polymer. So in the last few years, what we've been trying to attach to these polymers was like quite a variety of things. So we were missing Nutella, but like we'll try in the future, which is a great idea from Dimitris. So we've been playing with amino acids, fatty acid, cholesterol-like materials. So in that way, we can change the thermal properties, the amphiphilic balancement, so the self-assembling, and of course the architecture. So I will give you then, like in the next few slides, a series of proof of concept of how we use this functionalization, but also we can attach directly without any linkers some drugs. And also we use these two model drugs, methotrexate and mefenamic acid, to create some active polymeric prodrugs that can self-assemble in nanoparticles. And I'll give you like some ideas of how we use these final polymers as well. So what we thought was like, okay, we can create a library of polymers starting from the same backbone. So we can have like a statistical change of the materials. And so we wanted to prove if it's true that changing the chemistry of the polymers, we can improve the interaction with drugs. So a big chunk of the work, of the PhD work of Rashida, was to take all these polymers and make formulation with three really potent anti-cancer drugs, enoposide, etoposide, and toposide phosphate. So a really, really long story short, she prepared films of all the formulations, and she studied from these films the release and some chemical-physical parameters. And in that way, she screened the films, and she ended up taking, like, for each drug, one key polymers. And these polymers ended up in formulation in nanoparticles. And for example, for etoposide, the best polymers was this PGA modified with this really long steric chain. We could get nanoparticles, we can encapsulate the drugs, and we could have like a controlled release with time. And these, the change also of the change of the uptake of these drugs was translated in a decrease of the IC50 values in a 2D in vitro set of experiments. Moving on the possibility to predict at least the behavior of these materials in some in vivo, future in vivo experiments, Alexandra focused most of her time as a PhD here in Nottingham in studying some 3D spheroids models to understand how particles can penetrate through the spheroids. So she has been studying several commercial nanoparticles, but most of them they don't have like the properties she was looking for, because normally they are polymers like polystyrene with really high TG and are really rigid material. So our library of polymers could have given her like a better insight on studying on the properties of the polymers related to the penetration through the spheroids. So she started studying the two more flexible polymers we had, so the bare PGA and the pegylated version of that. And these two polymers have like TGs below minus 30 degrees, so at room temperature or body temperature, they are behaving like as a really nice nanoemulsion, and they are like quite flexible and can squeeze. And in fact, after only 4 hours, we can see that the particles are present in both in the rim, middle, and the core of the spheroids. However, if we attach our C18 fatty acid, what is going to happen is that the material becomes semi-crystalline and really rigid, and that it got stuck in between the rim and the middle of the spheroid, and takes over 24 hours to reach the center of the spheroids. However, because this is a quite versatile tools, we attached again we pegylated the polymer. The PEG chain break down the crystallinity of the materials, and the polymeric nanoparticles can get again through the different parts of the spheroids. And this is like quite promising to see how these materials can both interact with drugs in different ways, but they can also be studied and maybe used in the future in vivo experiments. As one of the main problems with all the aliphatic polyesters, they are always said that they are like biodegradable, but no one knows exactly which is the time frame of how it happened. So, and I had the pleasure to work with Sadie, and she spent lots of her time during her PhD, lots of efforts and lots of swearings against these polymers. We developed then at the end like a series of essays for the chain to see how it was degrading. But as it was mentioned a few times today, like the polymer is one thing, the conjugate is something else. So to mimic that, we produce some, for the sake that we can attach different things to this polymer, we attach the carboxyfluorescein directly to the polymer. And this can mimic both the presence of a drug and giving us ideas of how the material is behaving when it's not just the bare polymer. The nanoparticles were incubated with an enzymes, and according to the enzyme and the buffer, we could see that there was a different release with time. But anyway, in all the cases, the particle seems to be pretty stable without the enzymes, and there is like a burst effect when the enzyme is present. So there we have more details. We know that with more modification, this is different. But we have ideas that like polymers start degrading in the first 24 hours or less. And finally, just to give you like a quick overview of the model drugs we use so far, Deepak always been quite interested in solid dispersion, and so he was working on mefenamic acid, that is a anti-inflammatory drugs, highly crystalline, super difficult to dissolve in water. So what we did when we attached the drug to the polymer, the final polymer prodrugs was completely amorphous and was used to improve the interaction with other drugs. But in particular, what we could do, what we managed to achieve is that also the prodrug is self-assembling nanoparticles. And also in this case, in the presence of a lipase, the release of the drug is get a burst effect when an enzyme is added. At the same time, Kang was extremely interested on the effect of methotrexate attached to our polymer. We created different polymer with different amount of methotrexate. The particle could have been self-assembled at different pH, and they were pretty stable without any enzymes for different days. I mean, again, Kang in Thailand had lots of students, and they stayed along for the period we were studying for less, because we were just me and Deepak for the mefenamic. However, also in this case, once we added an enzyme, there was a burst release, hinting that the polymers is pretty unstable in presence of enzymes. And this was translated in a series of 2D and 3D cell culture experiments. And to cut a really long story short, also in this case, some of the polymeric prodrugs behave at the same or even better of the drugs alone, even against some resistant cell lines. And this is likely, again, due to the different mechanism of uptake. So I would like to thank all the collaborators that have been involved in this series of projects and this overview. And of course, my new group here in chemistry, the Audel group, and you for your attention. And thanks again, the organizer of this event.
Thank you, Vincenzo, for this very interesting talk. Let's check if we have questions. So we have excellent talk from Professor Cameron Alexander.
Thank you very much.
And I see two hands, so we can start with Thomas Moore. Thomas, are you able to talk or?
I think, can you guys hear me?
Yes.
Yes.
Okay, thank you very much for the talk.
But not hear you for now.
Can you hear me?
Yes, Thomas, I can hear you.
Okay. Thank you very much for the talk. I really was interested
meanwhile,
in this.
We wait for Thomas to eventually solve this connection problem. We can.
I can hear.
Okay, maybe it's back. Okay.
Okay. Sorry. So I really like this part about the nanoparticle penetration into the spheroid. It's a very interesting experiment. Is this purely related to the rigidity, stiffness of the polymer, or do you think there's also some kind of interaction between the polymer of the nanoparticle with the extracellular biomolecules or even the cell surface?
Yeah, Thomas, I mean, like you picked the problem here. Yes, because Alexander has been working on the model for a long time with polystyrene particles with different charges on the surfaces, and she was showing that changing the surfaces, she was getting through more or less. Unfortunately, we studied the PGA library a bit towards the end of her PhD. So the main insight we had was mainly the flexibility of the chain. And I can tell you that this is likely to be mainly that because the rest of the polymer is the same. However, it has been seen that like in a similar system without PEG, the same materials couldn't go through anyway. So there is a lot here to go through, but yeah, you picked the good one.
It's a very interesting problem to have, so thank you very much.
Thank you, cheers.
So we have maybe a question from Professor Cameron Alexander. Do you want me to read it, or do you want to connect? Maybe it's not able to, so I will read the question. So how much can you vary the links between the adipate groups? For example, by Dissulfate without changing the TG too much for particle self-assembly?
Yes, that is what we studied lately with PP, and we thought that changing the nature of the diodes, we were changing dramatically the TG, but with all the modification we had, we remained in below 0 degrees. So we are on the same, I mean, talking about like body temperature, we are all far below. Yeah, the difference will be maximum 10 degrees at the moment.
Thank you, Vincenzo. We have one last question from Claudia Conte.
No, Vincenzo. Just a curiosity, I know very well your work. You have done a very nice and funny presentation.
Thank you.
And I have just a curiosity, you have done a great, wide library of these kind of polymers, and you have studied the polymers several times in cell culture and 3D models. I think that now you should move in vivo.
Yeah, definitely. Just to mention that before I joined Martin and Cameron, my first postdoc here in Nottingham, they did something. There was like a study of like to try to convince AstraZeneca to get the polymer on board for some kind of excipient application. And so we know that the polymer, the bare PGA, it's not toxic in vivo on statistical studies on mice and rats. Now, but yes, yeah, but I agree with you, like we should do more, and it's time to move forward. And this is what I'm trying to do, like now, expand the screening, and then go maybe like in vivo or something like that. I mean, starting maybe like with a more 3D and then go ahead. But yeah, that's the way.
Okay.
Yeah.
Thank you, Vincenzo. Thank you very much.
So it doesn't look like there are other questions. So thank you again, Vincenzo, for your contribution. And we can move to the next speaker. We'll have Anna Maria Morelli from the University of Bari Aldo Moro with a presentation on improving liposome performance in oral delivery of hydrophobic molecules. Hi,
Can you see my screen?
Anna Maria.
Hi. Can you see my screen?
Not yet. Let's wait a couple of seconds. We don't see it yet.
No.
No. So let's check. We can check offline if there are problems with your link or something. Meanwhile, we can go ahead and have the next speaker. So we'll come back to you, Anna Maria.
Okay.
So we have now Beatrice Simonis from Sapienza University of Rome with a talk on design on novel liposome formulations for the delivery across a human blood-brain barrier model.
Good afternoon.
You, Beatrice, you can start.
So, good afternoon. First of all, I would like to thank you, the organized committee, for giving me the opportunity to present today. And my work is focused on the development of novel cationic liposome formulations for the delivery across a human blood-brain barrier model. So this brain barrier is a dynamic and complex interface between the blood and the central nervous systems. It acts as a selective biological filter, limiting paracellular and transcellular permeability and transport, and also prevents almost the totality of drugs candidates from reaching therapeutic concentration. So we can say that this extreme efficacy of this barrier is an obstacle for the treatment of brain disease. And for these reasons, the development of new drug delivery systems able to encapsulate a large amount of drugs across the barrier, reach the district at therapeutic concentration is still an urgent need. So in this nanosystems, such as liposomes, can be a very powerful tool. Thanks to their feature, they can be easily and properly tuned. They are low-cost nanosystem, usually show low toxicity, are easily to prepare and handle, and are able to deliver both hydrophilic and hydrophobic compounds. So the idea is to develop novel cationic liposome formulations. Of course, liposomes need to be functionalized to be able to cross the barrier, and cationic liposomes, so thanks to their positively charged surfaces, can easily interact with the cell membrane and enhance the uptake. So we prepare and characterize different liposome formulations, and on most promising ones, the bioelement was done in collaboration with the ARBM. In particular, liposomes were marked with a fluorescence probe for uptake experiment on brain endothelial cell monolayer, and for transport experiment, liposomes were loaded with a quinolanic acid, that in this case was used as a tracer because it's unable to cross the barrier. Of course, the final goal is to achieve liposome formulation stable, not toxic, able to cross the barrier, and able to loading a high amount of drugs. So prepare and characterize different liposome formulation composed of natural phospholipid, PPC, and cholesterol, in mixture with cationic diastereomeric Gemini amphiphiles, SS or meso, that should promote the crossing of the barrier by absorptive-mediated transcytosis, or in mixture with a monosilate lipid that should favor the crossing by carrier-mediated transcytosis, in particular group 1. All liposomes were prepared according to the lipid film hydration and extrusion protocol, and all liposome formulations were characterized in terms of size distribution, polydispersity index, and zeta potential by the LS measurement. So here we reported the characterization of liposome containing the 2 Gemini surfactant SS and meso. Here we wanted also to investigate on how the different stereochemistry of these 2 Gemini can affect the physicochemical feature of the final liposome formulations. So from the DLS results, we observed that we achieve a narrow side distribution for all liposome formulation with hydrodynamic diameter in a range between 130 and 180 nanometer. We also evaluated the stability of this formulation over a period of 7 days. After that, the formulation containing the cationic Gemini SS showed an increase in size, compared with the same formulation containing also the PEG, that not shows any tendencies to aggregate, so probably the presence of PEG preventing the aggregation phenomena. On the other hand, the formulation containing the other diastereomeric Gemini meso remains stable over a week. These formulations were characterized, and in this case, the lipid films were hydrated with a quinolanic acid solution, and the capability to entrap and retain the quinolanic acid was assessed by HPLC measurement. Also, in this case, from the DLS results, we achieve a good hydrodynamic diameter and a suitable quina concentration for the biological test. The formulation containing Gemini SS and Gemini meso also are able to entrap a large amount of quinolanic acid compared to the formulation containing only the PPC and cholesterol. We also evaluated the release of quinolanic acid over time, and the leakage of 50% was observed after 8-9 hours from the preparation of the samples. However, the presence of Gemini SS and meso involves a slower release. For this reason, the samples were prepared immediately after use for the biological test. In addition, in order to obtain a doubly functionalized formulation, we developed a formulation composed of cationic Gemini SS and a ligand able to bind the GLUT1, so in this case, a monosilate lipid. Also, in this case, the formulation was characterized in terms of particle size, polydispersity index, and zeta potential by the LS, and the quina concentration by HPLC. We also evaluated the release of over time that follows the same trend observed for the previous ones. So, as concerned the biological experiment in collaboration with the IRBM, liposomes were marked with a fluorescence probe for uptake experiment on a brain microvascular endothelial monolayer derived from human pluripotent stem cells. Instead, for permeability experiments, liposomes were loaded with a quinolanic acid that was used as a tracer because it's unable to cross the barrier, and we evaluated the permeability of the free quinolanic acid compared to the permeability of the quinolanic acid loaded into liposome formulations. So biological experiments were done on the most promising formulations, so formulation containing Gemini SS, Gemini meso, and monosilate lipid. The uptake experiment was investigated to temperature 37 and 4 degrees. So here we can see the images recorded under the fluorescence microscope, and we can see red the junction of the membrane, in blue the nuclei, and in green is highlighted the fluorescence of the lipid probe. Following the internalization process at 37 degrees, we can see that the formulation containing only the PPC and cholesterol does not interact with this brain monolayer, compared with the other two formulations containing Gemini SS and the formulation containing both Gemini SS and monosilate lipid, in which the uptake is higher. So this suggesting that the presence of this Gemini or the presence of this monosilate lipid promotes the internalization process. On the other hand, following the internalization process at 4 degrees, we can see that again, the formulation containing only the PPC and cholesterol does not interact. And for the other two formulations, the uptake was reduced. So that this suggesting the possible existence of an energy-dependent transport. Similar results were also observed for the other two formulations, the formulation containing both Gemini SS and PEG, and the formulation containing the other diastereomeric surfactant meso. We also evaluated the permeability of this liposome formulation on in vitro barrier model compared with the free kinase. The model is a culture of brain microvascular endothelial cells into the upper compartment and a human astrocytes into the lower compartment. The monolayer integrity of this barrier was confirmed by measuring the high value transendothelial electrical resistance and by measure a paracellular market that was used as control because it's unable to cross the barrier and was measured during the whole experiment. So samples were loaded into the upper compartment that represent the blood, and after 1 hour of incubation were recorded from the lower compartment that represent the brain. So results highlights that the permeability of kinase loaded into the formulation containing only the PPC and cholesterol is comparable to the permeability of the free quinolanic acid. And on the other hand, for the formulation containing AMYC and Meson, in this case, the permeability significantly increased compared to the formulation containing only the PPC and cholesterol, and also compared to the free quinolanic acid. In particular, the formulation containing the AMYC show a significantly higher permeability. Instead, the other formulation containing the mannose-late lipid, in this case, the permeability is undetectable. So probably this mannose-late lipid at first enhanced the uptake by ligand recognition, but after is involved in some cellular trafficking, probably landing in lysosome degradation. So in conclusion, different liposome formulation functionalized across the barrier and loaded with quinolanic acid were prepared and characterized. Formulation containing Gemini SS and Meso or mannose-late lipid were also evaluated on a brain endothelial monolayer. And the results shows that the presence of this Gemini or the mannose-late lipid promotes the internalization process with the brain monolayer. We also evaluated the permeability of this formulation on an in vitro barrier model using quinolanic acid as a tracer. And the permeability of quinolanic acid loaded into liposomes containing AMYC and Meso significantly increased compared to the free kinase and compare also the formulation containing only the PPC and cholesterol. In particular, the formulation containing the AMYC show a significantly higher permeability. On the other hand, the formulation containing mannose-late lipid that at first seems to favor the internalization process, in this case show a very low permeability across the BBB model. So in conclusion, we can say that the presence of this Gemini and the different stereochemistry can play a role by influencing the physicochemical and biological properties of the final liposome formulations. So I want just to thank you, all the research group who collaborated to this project, IRBM Group, CNR, and Sapienza Group, and also for the fundings, CNCCS and Regione Lazio, and of course all of you for the attention.
So thank you, Beatrice, for your contribution. Let's see if there are questions. I don't see question in the chat or in the any raised hand. So I would like just to know if you consider also the evaluation of the stability in physiological fluid, if you have any clue about it, if you are working on that, or if you are planning of doing it.
We are planning to do it because it's a very critical point. And we want to also study the behavior of this formulation with and without the presence of PEG. So, but we are working now on that, so I don't know.
Okay.
I'm waiting for.
Thank you. I don't think there are other questions. We can try again with Anna Maria Maurelli.
Yes.
Can you share the screen, Anna Maria?
Yes. Can you see my screen now?
So you can start your presentation.
Okay. So I am Anna Maria Maurelli, and today I will present you my work that's about the use of liposomes for the oral delivery of antioxidant molecules. So these are the topics that we will explore today, starting from the theoretical background and then the goal of the study and the results obtained. So liposomes are widely used nanocarriers in biomedical and food technology applications, and they are really similar to the biological membranes since they are composed from a phospholipidic double layer, and they can easily interact with cells. Moreover, they can encapsulate both hydrophilic and hydrophobic cargos. So they have been exploited for different applications, which go from drug and nutraceutical component delivery, stimulus responsive delivery, in gene therapy, and so on. Different studies demonstrated as curcumin has antioxidant, anti-inflammatory, and antimicrobial properties. However, it has a low solubility in water, instability at pH higher than 7, and a rapid metabolism. So we tried to encapsulate it into the lipidic bilayer of liposomes in order to overcome this limitation and exploiting its hydrophobic nature. However, liposomes themselves are not able to cross the gastrointestinal tract since they are destroyed into the stomach. So the goal of our study has been to adapt liposomes for the oral delivery of antioxidant molecules, and then investigate their cytotoxicity and antioxidant activity, and realize an in silico optimization to find the values of lipidic and curcumin concentration, which gives the best performance in terms of encapsulation efficiency and loading capacity. We prepared our liposomes with curcumin mainly in 2 ways, with the micelle to vesicle transition method and with the extrusion method. The first passed through the formation of mixed micelles, and then the detergent deflation induces the formation of liposomes. And with this method, we can obtain liposomes around 30 nanometers in diameter. The second method was the extrusion method, in which first polydisperse and multilamellar vesicles are obtained, and then using membranes with a defined porosity of 80 nanometers, we obtain liposomes. In order to adapt these liposomes for the oral delivery, we cover them with the Udragit 100, which is a copolymer of methacrylacrylate and its methyl ester. And this is a stable coating at acid pH and then dissolves at neutral and alkaline pH. We realize this coverage with a pH-driven and organic solvent-free method. Briefly, by adding the liposomes alkaline suspension into an acid solution, the polymer precipitation around the vesicles occurs. Then we evaluated the interaction of the cells with of the liposomes with the CACO2 cells of the intestinal cell line. The MTT test showed that there is no significant difference between the control and the interaction of cells with liposomes, curcumin loaded liposomes, and the system with the Udragit, demonstrating the safety of our samples. Then, through a flow cytometry analysis, we evaluated the uptake of the liposomes with cells before and after the treatment of the sample with the trypan blue, with the quenches the extracellular fluorescence. In these images, we can see a lack of a difference between before and after the treatment with the trypan blue, and also a good cellular uptake of our systems. Then we evaluated the antioxidant activity of the system in vitro, performing an assay based on the use of dichlorodiacetate fluorescein. Briefly, in presence of ROS, this compound is converted into DCF, which is a strongly fluorescent compound. So the fluorescence of this compound can be correlated to the antioxidant activity of the cells. This test has been done in using an oxidative cell stress in presence of hydrogen peroxide. And we saw that concentration of hydrogen peroxide higher than 0.5 millimolar were toxic for cells. So we fixed the concentration at 0.1 millimolar, and we evaluated the antioxidant effect of pure liposomes, curcumin-loaded liposomes, and Udragit curcumin liposomes. and we saw a decrease of the fluorescence, so an antioxidant activity in presence of the curcumin. With and without the Udragit after its dissolution. In presence of the coating at acid pH, systems does not have any antioxidant activity, demonstrating the protection given by the polymer to the systems. Then we make a comparison of the curcumin entrapped into the liposomes with the curcumin in ethanol, performing a decoloration assay based on the neutralization of the radical cation ABTS+. The antioxidant activity in this case was evaluated in terms of TIAC, which is Trolox equivalent antioxidant capacity, and using Trolox as an antioxidant standard compound. Also, in this case, we can see that curcumin in liposomes has the same antioxidant activity that the pure curcumin in ethanol, demonstrating as liposomes does not alter the antioxidant activity of this molecule. In a second step, we realize a double polymeric coating around our liposomes, adding also the PEG. In this step, we used a lipid with the head modified with the PEG group at 7% of the total mass of lipids. This percentage assures the presence of the PEG in the so-called brush-like configuration around the vesicles. The PEG on the surface of liposomes stabilizes spherically the vesicles, prevents their fusion, makes them resistant to enzymatic and bile salt degradation, makes them invisible to the immune system, and mucus penetrating. In the pathological condition of the gastrointestinal tract, the mucus layer that covers the epithelium is increased compared to the norm. Therefore, the ability of the vesicles to cross this layer is a desirable condition. Then we performed an in silico optimization of the encapsulation efficiency and loading capacity, varying the quantity of curcumin concentration and lipid concentration in the formulation. In this case, the quantity of curcumin trapped inside these systems has been evaluated in a spectroscopic way, exploiting its peak of absorption at 420 nanometers. We can see from the graph that the loading capacity is maximized near the highest value of curcumin concentration and the lowest value of lipids concentration, while the opposite has been found for the encapsulation efficiency. From the overlap of the two contour plots, we can see that the concentration of lipids at 1 mg/ml and the curcumin between 17.5 and 18 micromolar is the one that maximizes both the encapsulation efficiency and loading capacity. Finally, TM and DLS analysis show that before the coverage, liposomes were around 80 nanometers in diameter. After the coverage, Udragit forms structure with clusters of liposomes, which are then released after the dissolution at neutral pH. And so releasing them in the conditions in which they will be absorbed in vivo in the colon. Thank you for your attention.
Thank you, Anna Maria, for your presentation. I see Claudia typing a question. So have you checked the PEG on the surface? So the thickness of the shell or?
Not yet. In literature, we read that the PEG group occupy around 5 nanometers or 10, but we didn't see any difference.
Okay, thank you. Now we have another question from Thomas Moore.
Hi, thanks. Can you hear me?
Yes, we can hear you.
Thank you for the talk. I was curious, one of the last slides you showed there's this kind of
Can you hear Thomas?
No.
Thomas, we lost the connection. I don't know if you can hear us. No. Let me check if meanwhile we have other question. Yes. Just 1 second, we'll see if we can type the question. I don't see text right now. Ah, maybe he's back. Thomas, can you hear us? If you can also type the question if it's easier. Okay. So he was curious to that you see this opposite relationship between the lipid concentration and loading capacity or encapsulation efficiency. Do you know what is happening here physically?
Just a moment. Yes, maybe I can share again the slide. Can you see it?
Yes, we can see the screen.
Okay. Yes, we are still studying these data. We acquired them in the last period. So the opposite relationship between lipid concentration and loading capacity. Oh, yes, encapsulation efficiency and loading capacity are evaluated in a different way. Encapsulation efficiency is based more to the percentage between the curcumin into the system, entrapped into the system, and the one that we put at the start, while the loading capacity is more correlated with the weight of the nanoparticles, so with the lipid concentration.
Okay.
I don't know.
Thank you, Anna Maria. And with this, we reach the next coffee break. So we are some minutes in late. So I will say that we connect again 10 minutes after 4 to following the afternoon session. Thank you to everyone participating up to now, and we'll see you in 10 minutes.
Sto facendo le prove. Come si fa a rimuovere questo coso? Oh mio Dio, come si fa?
In alto a destra c'è vista.
Fiamma. Esci da schermo intero.
Sei proprio una boomer. Ehi,
Che vuoi?
questa qua.
La odio, aiutatemi. Mo questo come devo dire? Vabbè, non dico niente. Non dico, vedete la mia presentation. See you my presentation.
Giulio, sì.
Disattivate
Can
l'audio,
you
per
see?
favore.
Che
Okay, welcome back. Now we are going to start with the last session of the day. That is the last session of the conference as well. The next talk is a sponsored talk by the editor Wiley. The title of the talk is Current Challenges in Chemistry Publishing from Francesca Rita Novara, Editor-in-Chief of Chemistry Europe Journal. Francesca, you can share your screen and start the presentation.
Hi everyone, I'm going to look for the presentation.
Yes.
Can you see anything?
No, not yet.
Ah, okay.
In the meantime, I would like to thank, of course, Wiley to support us for the conference, for your contribution.
We are really happy to be here, and it would be also really good if I could share this talk with you.
Thank you very much, Francesca.
Okay, so I'm trying to share the screen.
Yes.
Haha, maybe I can do this. Can you see this?
Okay, yes, yes.
Okay.
Great.
All right.
You can enlarge the screen with the...
Yeah, I really should have practiced this when you offer it. Okay. Hi.
Okay.
So, thank you very much for having me today. Thanks for the organizers, and thanks Stefano for giving us the opportunity to be present. I heard lots of great talks this afternoon, and I'm very happy that I could be a part of this today. So we only have 10 minutes, so we're gonna make this short, and I'll try that I can be useful to you. My name is Francesca Novara, and I am a scientific editor. This means that my job is to make it easy for others to communicate, for scientists to communicate their results and their research, and to do this in the most efficient, smooth way, and also to communicate it to the most appropriate audience. And this is what we do, prevalently at Wiley. I do this with Wiley, which is a very old publisher. It has been in the publisher business for over 200 years. It started as a little printing shop in Manhattan in the US, and now it has expanded globally, and it has offices all over the world. I think we are over 5,000 employees. And my office is the one based in Weinheim, which is a little bit south of Frankfurt in Germany. In Weinheim, we are over 400 people, and it is a very diverse and interesting community. People come from all over the world to do this job. And we have book services there, we do marketing activities, and we also publish scientific journals in Weinheim. The journals I'm involved with are the Chemistry Europe journals. Probably you've heard of these journals before, but let's start from the beginning. What is Chemistry Europe? Chemistry Europe is a consortium of European chemical society that joined forces in the middle of the 90s, and they decided that they wanted to publish together excellent chemistry journals. The Italian Chemical Society was one of the first to join, and some of the oldest journals we have came also from old Italian journals, such as Annali di Chimica, for instance. Chemistry Europe journals are committed to the best standards in scientific publishing, and until now we have 19 journals that are part of this family. For these conferences, 3 of them have been involved as sponsorship, the one that matched the most with the content of your symposium. They are our newest launch, Analysis and Sensing, the more established ChemMedChem, and the open access journal Chemistry Open that I am responsible for. So what do editors look like, and what can they do for you? So this is a photo of the Chemistry European Journal editorial team that I am part of and have been for almost 10 years. So that's how we look. We are quite international and not always this smiley. And what can we do for you, and what do we do? The typical education of an editor in our team is to have a PhD in chemistry. This also I'm happy to tell you, because you're all chemists, you're at the beginning of your career, and we like to support young researchers with their papers, but also later on in their career if they maybe want to become an editor. This is I can only say that this is an excellent opportunity to keep being involved in what happens in science without doing it actively, should this not be something for you. Mostly we are involved with the evaluation of the manuscripts that are submitted to our journals. So peer review and production tasks. Then we are involved in the monitoring of the latest scientific developments. So we attend conferences such as this one. We are involved in the acquisition of manuscripts because we always want to make sure that the journals have the best content possible and that the content is reflecting the latest development in a given field. So this is a little bit the most important task for us. And this is a very complicated slide that maybe is not right for a Friday afternoon, but just at a glance, this is the editorial process from submission to the article until its final appearance online in a journal. Yes, if you're interested in something more in depth about this, maybe you can send me an email. I don't think it's something that we should go into depth for now, because even though it was not allocated time for questions for this little session, I think maybe we should make time for it, seeing that maybe some of you have questions to ask. One important thing that I wanted to share is that 70% of our rejections are then referred to another journal in our portfolio. And this often also happens after the first submission. I know it's quite a disappointing moment when the submission is not deemed suited for the journal that was chosen. However, it is in our interest to find the most appropriate recipient for every paper. And very often this is a successful process. And so, yeah, take advantage of every transfer offer. And I think, yeah, then what I want to say that also we have virtual events of our own. So you can check our Chemistry Europe Hub to see what we're up to and what latest events are happening for us. And with this, I wanted to terminate the overview of the Wiley journals. And if we still have time, I wanted to move to one aspect in particular. As my talk is titled Current Challenges, I believe that one of the most important challenges is the transition to the open access publishing model. And I strongly believe that the future of scientific publishing is open, and open not just in the sense of open access as a publishing model, but also open science as a way of conducting science. I believe in this model, and I think the advantages are more complex than just considering that the paper can be read everywhere. But anyway, so yeah, in my opinion, and I hope we still have time for this, it will be only 4 more minutes. The idea of open is very connected to the idea of freedom. For example, when one thinks about open access, one thinks, okay, I pay something or someone pays for me something and then it's free. But it's not just free in that it's like in the example here is a free beer, but it's free also like gives you the possibility to use that content again. For example, if you think about it, there are 2 ways to consider the word free. On the one, meaning of the word, it's for free, as in gratis, so you can see something for free. But in the other understanding of this word, it's free in that you can do something with it. You can build upon it, such as in this case with the Gioconda that has been used to produce marketing tools. So freedom from the payment to read or freedom for reuse. And in the open access world, you have both of these meaning that are active. And this not only for the publishing, but also for the ways of doing science. For example, if you conduct your science in an open manner, then all the experiments are free for everyone else, for all the other scientists to be used. So this is more the less obvious meaning. Obviously, there is a paywall, and I'm there is a paywall for publishing in this open access way, and yes, everyone is concerned about this, and I just wanted to let you know why there is this paywall. So for example, all the editorial work on our side, including my job, is part of this paywall. Then the management of the peer review, the software that we use, for example, to process the manuscript, but also to make sure that the manuscripts that we handle are ethically correct, and also the costs of the production of the manuscript itself. So this is all things that have a cost, and by paying that cost, we can make sure that the product that you have in the end is a good product. So in the usual model, which is the traditional model, the non-open access model, this value comes from the readers upon paying the subscription. And in the open access model, this sum comes from the funders. So, sorry, this is a bit convoluted. On the one side, you have the authors. On the other side, you have the readers. And I'm not sure if I still have time to discuss something else. I think I will stop sharing now, and I will give you an opportunity to ask questions, if there are any.
Thank you, Francesca. Thank you very much. There is any question from the audience? No, nothing in the chat. No question. No, Francesca, no question.
Okay, that's fine. If you have any questions to the open access journals or to any of our journals, please reach out to the editorial offices pages, and or if you're interested in an editorial talk with more time, then we are happy to participate. And it has been a pleasure to sponsor.
Thank you very much. Thank you again for your support. So, no, just a second. We have Stefano.
I have a question. Sorry,
okay,
Claudia. Sorry. Thank you, Francesca, for this presentation. I have a quick question regarding the role of a young researcher. So, I mean, sometimes this kind of open access journal now requires some fees, no, to be published more than the other journals. So even if sometimes there is some special offer, no, due to specific reasons. So especially for young researcher, for young group leader, that sometimes they don't have a lot of money to pay for a publication. Is there any form that I don't know, of credit that Wiley can allow or not? Or
yes, where, for example, in Italy now there is the transitional deal in that the fee is paid by the institution almost upfront. So all the Italian university can publish open access in our journal at no further cost to the author. And this is only an example that the society is moving in this direction. The idea behind open access is not that the author needs to pay out of his own research grant, because this is simply not sustainable. However, at Wiley, we also have the possibility to waive the papers for researchers that cannot afford it, if they want to publish in a specific journal. Otherwise, there is also all the other journals where one can publish in. Also for publication in those journals, there is a transaction. It's the subscription.
Okay.
So there is always a transaction. It depends on which side, like I was saying earlier. But there are funds for promoting open access publication for young researchers. Also at Chemistry Open now we will inaugurate at the beginning of next year an early career advisory board, and everyone on that board can also publish for free. So yeah, we are trying to use these initiatives also to promote it among those that have countries with low income, or also people at the beginning of their career that don't have this kind of budget.
Okay, thank you. No, because of course the open science and culture now will be I think very important for us, but sometimes there are some barriers for this. So it's very nice to hear that, especially for young researcher, but not only young researcher, there is the possibility, thanks to our institution, thanks to our scans, to submit this. Thank you very much.
And also, as a last point for the Chemistry Europe journal, our revenues are also given back to the societies in form of royalties. They are so-called royalties, but it's money what it is. So also this is part of this money is used to support the activities of the societies.
Thank you, Francesca. Thank you, Wiley. Thank you.
Bye.
Thank you again. Bye, Francesca. So now let's move to the first oral communication of this last session. Is from Sofia Parrasia from University of Padua. Sofia, okay.
Hi.
Are you connected? The title is Brain Delivery of Mitochondriotropic Inhibitor of the Potassium Channel Kv1.3, Conjugation with Peptide Targeted Blood-Brain Barrier. Okay, we can see your screen. You can enlarge your screen. Okay, perfect, great.
Okay, thank you very much. So good afternoon. Today I will share with you most of my PhD work that was focused on the brain delivery of a promising anti-cancer drug that acts as an inhibitor of the voltage-gated potassium channel 1.3. So to briefly introduce the background of the project, Kv1.3 channels are expressed both at the plasma membrane and mitochondria level. This different subcellular localization accounts for different functions. Indeed, the inhibition of the plasma membrane channel leads to a decrease in cell proliferation, leading to a cytostatic effect, while the inhibition of the mitochondrial population of the channel stimulates apoptosis, leading to cell death. These channels have been studied in the cancer field, and it has been found that they are overexpressed in some cancers. Some of these are listed here in this picture. And what is of particular interest is that glioma, pancreatic ductal adenocarcinoma, and melanoma, for example, are very difficult to treat also with standard approved therapies. So Kv1.3s have been studied as a target for cancer therapy. So given the relevance of the inhibition of the mitochondrial population of the channel to trigger apoptosis rather than the reduction of cell proliferation, PAP1, which is a membrane permeant inhibitor of this channel, was modified here in Padova by the addition of a triphenylphosphonium moiety in order to achieve the mitochondriotropic derivative PAP-TP. This strategy to add a triphenylphosphonium moiety is a common strategy that exploits the different membrane potential between cytoplasm and mitochondrial matrix. In this way, PAP-TP, once administered, can accumulate in mitochondria and inhibit preferentially the mitochondrial population of the channel, leading to apoptosis. PAP-TP was successfully studied in vivo in some cancer models such as PDAC and melanoma, and it turned out to be very effective. As you can observe in this graph, it was able to reduce tumor weight in case of PDAC and tumor volume in case of melanoma. PAP-TP was also tested in glioma cell lines, and it turned out to be very effective in the activation of apoptosis. But when tested in vivo in an orthotopic animal model of glioblastoma, it turned out to be completely inactive. This negative result was then explained by pharmacokinetic studies showing that PAP-TP is completely unable to cross the blood-brain barrier. Among other strategies, a common strategy to achieve the BBB crossing of drugs is the use of peptides able to cross the BBB. And one of the most promising in this field is AngioPep2. AngioPep2 is able to cross the BBB thanks to its affinity for the LRP receptor. This receptor is expressed at the brain capillary endothelial cell levels, but it is also expressed by glioma cells. And in context of brain tumors, it is very important because the use of AngioPep could allow the accumulation of drugs in the tumor. Of note, AngioPep2 also reached the clinical phase 1 for the delivery of paclitaxel. So coming to the aim of my study, based on the inability of PAPTP to cross the blood-brain barrier, our idea was that the conjugation of PAPTP with AngioPep could allow its brain delivery. The model was designed as a prodrug that should be stable enough to allow the crossing of the BBB, but at a certain point should be able to release PAPTP, which should act as anti-cancer drug. Here I reported the structure of our construct. You can observe here PAPTP that was conjugated through a bioreversible carbamate bond that should allow the prodrug feature of this construct. It was not possible to directly conjugate the carbamate to the triphenylphosphonium moiety of PAPTP because it gave a compound completely unstable. So we had to modify PAPTP with the addition of a spacer. Then isoleucine was used as an intermediate amino acid to allow the coupling with AngioPep. AngioPep synthesis was performed by peptide solid phase synthesis, growing the chain toward the N-terminus. When the desired sequence was obtained, the N-terminus was deprotected and conjugated with the PAPTP linker isoleucine. Then all the amino acids were deprotected and the conjugate was unlocked from solid phase. As I said before, PAPTP was modified by the addition of this linker that was added to the triphenylphosphonium moiety because this part of the molecule, which is composed by the psoralenic moiety and the phenosybutossi arm, could not be modified because it's very important for the inhibition of the channel. So the first point was to evaluate whether this modification could alter the cytotoxicity profile of the compound. So we performed an MTS assay on neuroblastoma cells, and we found that the modification of PAPTP did not alter the cytotoxicity. So we went on and we evaluated, before starting with in vivo studies, the blood stability of this conjugate. This was done because, of course, being a prodrug, it should be stable enough, but it should also be able to release the anti-cancer drug. So the conjugate was incubated with mouse blood, and we followed the release of PAPTP linker by HPLC analysis. The results show that half of the conjugate was hydrolyzed within 2 hours. So we concluded that the conjugate was stable enough to proceed with in vivo studies. At this point, so we administered the mice AngioPep2 PAPTP by intravenous route, and we evaluated the brain accumulation and the liver metabolism of the molecule. The time point that we chose were 15, 30, and 60 minutes. The results showed that AngioPep2 successfully delivered PAPTP to the brain. As you can observe in the graph, we found the conjugate at both 15 and 30 minutes after administrations. On the other hand, after 1 hour from administration, the conjugate was only present as traces that were under the quantification limit. As you can observe in the table where I reported the concentrations of the molecule found in the brain, these are in the low micromolar range, and these concentrations are probably too low for therapeutic application, but comparing the results obtained found in the literature, these concentrations are in line with what is reported concerning AngioPep2 ability to deliver drugs to the brain. Then we moved to the evaluation of liver metabolism, and we found that the main metabolites were PAPTP linker and PAPTP linker plus the first few amino acids of AngioPep2 chain. We also quantified the metabolites, and if for PAP-TP linker we did not find any significant increasing or decreasing trend over the time, for the other metabolites, we found a significant or close to significance increase after 1 hour. If you remember, after 1 hour, the conjugate is no more detectable in the brain. So we can hypothesize that this strong metabolism in the peptide chain could be responsible for the loss of angiopep2 recognition at the level of its LRP1 receptor expressed in the blood-brain barrier. In the last minute, I would like also to show you another approach that we explored that was based on the conjugation of PAP-TP with another peptide, which acts as a cell-penetrating peptide, exploiting a specific mechanism for the crossing of the BBB, and it mainly exploits amino acids with a lot of positive charges. We performed the same experiments with that PAP-TP, and we found that it was very, very stable in blood, even more stable because less than 20% of PAP-TP linker is released within 4 hours. But the brain delivery was comparable with that observed for angiopep2. Unfortunately, we experienced a strong toxicity, as mice need to be sacrificed after more or less 15 minutes from administration. So we tried to investigate this toxicity, and in the literature we found that one of the causes of toxicity for peptides with positively charged amino acids could be hemolysis. So we performed an hemolysis assay where the conjugate was incubated with the mouse red blood cells at different concentrations, and the release of hemoglobin was followed by spectrometric analysis. You can observe that that PAP-TP at the concentration that were found in the brain that are much less than what is found at the beginning, at the moment of an intravenous administration, is already hemolytic, causing an hemolysis of 8%. So we can imagine that that in the moment when we administer intravenously this compound to the mice, the hemolysis is much greater. On the other hand, angiopep2 PAP-TP confirmed to be safe and not hemolytic. So to conclude my talk, these results represent the first evidence of PAP-TP brain delivery, confirming that angiopep2 is a successful strategy for PAP-TP brain delivery. And this is also very important because it puts the basis for the exploiting of this very powerful anti-cancer drug for the treatment of glioma. The drawbacks is that the brain concentrations reached maybe are too low, but this could be improved, and this is what we are planning to do using a glioma mouse model. This could be an advantage because, of course, there is an enhanced permeability and retention effect caused by the presence of the tumor that alter the permeability of the BBB. And also, the concentrations could be improved thanks to the expression of the LRP1 receptor by glioma cells that could attract much more compound. With this, I would like to thank the scientific committee for giving me this opportunity, my supervisor Mario and Lucia for their guide, and our collaborators on the Chemical and Biology Department of the University of Padova for all their support. And thank you for your attention.
Thank you very much. Very nice talk, very nice presentation. There is any question from the audience? Let me check in the chat. No. I have just a curiosity. If you have tried different doses of your peptide, And if you have found
Yes, we tried.
the basis of the doses, if the percentage that reach the brain is different relating to the dose.
Yes, to be honest, we tried different doses with the TAT-PAP-TP because we started with an higher doses, but given the toxicity, we went back to the 5 micromolars per kilos. With AngioPep2 PAP-TP, we did not increase the dose because we were afraid to find some toxicity. So we used these concentrations because these are the concentration for PAP-TP that were effective in other models. So we did not went up.
Limit the toxicity. We have a question from Gabriella.
So I actually have 2 questions. First of all, if you have any idea of the mechanism of crossing the blood-brain barrier for your conjugate, and also if you have any idea of the biodistribution to other tissue. So you see that your conjugate is able to reach even a small concentration, but you are able to reach the brain. But then what is the distribution in other tissue like kidneys, liver, and so on?
Yeah, we evaluated the distribution to kidney, spleen, and heart, and the compound, of course, is distributed. We also found the same metabolites that we found in the liver, but in much less concentrations. So there is no particular accumulation in organs. About the mechanism for the BBB crossing, we think that the mechanism is given by the peptide. In the case of AngioPep2, there is the affinity for the LRP receptor. So the conjugate should attach to the receptor and then translocate. We also performed in experiments on bilayers of phospholipids to be sure that the compound did not alter in a specific mechanism the permeability of the BBB, but this is not the case. So we are quite sure that the mechanism is dependent on the receptor.
Thank you.
Thank you.
We have in the chat a question from Cameron. I don't know if Cameron is able to switch on the camera. Otherwise, I'm going to read how long was, for the TAT peptide, how long was the peptide sequence? Was it the short peptide version?
Yes, this was the short peptide version. Okay. Yes, it was the short one.
Okay. Thank you very much.
Thank you.
We don't have any further questions, so thank you, Sofia. Now it is the time of Professor Manish Kumar from Madhya University in India. The talk is based on the development and optimization of drug-loaded nanoemulsion system by phase inversion temperature method using a box bank can design. Manish Kumar is online, okay.
Thank you very much.
If you can share your screen.
Yes, thank you. You can see my screen?
Yes.
Thank you.
Perfect. Okay.
Thanks for giving me the opportunity to present my work. It is entitled as Development and Optimization of Drug-Loaded Nanomolson System by Phase Inversion Temperature using box Began design. So I start with the introduction. The nanomolson system are the lipid-based drug delivery system. They are very good in increasing the solubility and bioavailability of hydrophobic drug. Nanomolson system are the colloidal dispersion of two immiscible liquid, and one liquid is dispersed into the another, and the droplet size of one phase was below 200 nanometer. Another advantage of nanomolson system that they contain the triglyceride oil, which favors the lymphatic transport of loaded drug, which very good in enhance the bioavailability by avoiding the first pass metabolism. So there are different techniques for formulating nanomolson system, high energy and low energy method. High energy method generally requires sophisticated instruments, so they are costlier. The objective of our investigation is to develop a nanomolson system using the low energy method, which is phase inversion temperature method, which is simpler and cost effective. In the phase inversion method, the phase inversion is induced by varying the temperature. We can see that initially the system is oil in water emulsion. When we heat it above the phase inversion temperature, it converts into the phase inverse water in oil emulsion system. Phase is inverse through the bi-continuous microemulsion, and then we cool it immediately, and we induce the quenching of temperature. Due to this, there is sudden drop of temperature, and again water in oil emulsion convert into the oil in water nanoemulsion. This create a great turbulence in whole system, and due to that turbulence, nano droplets are formed. So with material, I used Banidipine. It is a anti-hypertensive drug, and it have the low aqueous solubility, high first pass metabolism, and its bioavailability is also affected with pH of gastric fluids. So for the optimization of nanomolson system, I used the Volkswagen experimental design. And in this investigation, I use optimized oil phase. I blend different long chain triglyceride oil, medium chain triglyceride oil, and essential oil to develop the stable nanomolson system. So I start my experimental work with the solubility. In long chain triglyceride, the maximum solubility is found in oleic acid. And with essential oil, we can see that cinnamon oil are good in solubility. It shows highest solubility about 85.46 mg per ml. Among surfactant, twin 80 shows highest solubility. So using this different oil, which are showing the highest solubility, I optimized the oil phase. For a stable nanomolson system, oil should be solubilize the drug in required quantity, should easily emulsify, should enhance the lymphatic transport of the drug, and should have the optimum stability. To serve this all purpose, I have used, I blend different LCT, MCT, and essential oil. These are the four composition I used in different proportion. And the selection of the proportion of different essential LCT and MCT oil, I conduct the ease of emulsification test by using 30% of oil phase and 70% of S mix, that is twin 80 and glycerol and 3:1. Then 1 ml of this mixture was added in 100 ml of distilled water, and time taken for emulsification and transparency is noted. We can see that the oil blend OB3 and OB4 shows transparency, so they are good in emulsification ability. But we use OB3, 40% of cinnamon oil, 30% of oleic acid, and 30% of MCT oil as our oil phase, because they contain higher proportion of LCT oil. The oil phase which contain higher proportion of LCT oil generally are stable, and they are good in enhancing the lymphatic transport of drug. So with this oil, I first conduct the compatibility study with FTIR. I take the spectra of pure Banidipine and Banidipine with excipient. The characteristic peaks of the Banidipine retained in excipient. So my drug was compatible with the excipient. Then with finally, I used drug with optimized oil phase. 280 was selected as surfactant, and in that investigation, I used glycerol as a co-solvent to investigate its effect on properties of drug-loaded nanomolson system. For optimization, I used 3-factor 3-level Volkswagen design. Total 15 formulations have been made. These are the independent variable, which are the amount of oil and surfactant glycerol. Dependent variable was droplet size and percentage transmittance. These are the total 15 formulation composition. The amount of oil, surfactant, and the pro-solvent has been given in the table. I start my formulating with determining the PIT, phase inversion temperature method. It is the temperature at which emulsion system got turbid. At this temperature, the non-ionic surfactant, which is 280, inverse their curvature. So phase inversion occurs at this temperature. So it was found to be 65-degree temperature. So for formulating nanomolson system, I take oil phase and S-mix and I heated it at 75-degree temperature, and I added the water phase at 75-degree temperature with stirring at 300 RPM. Then whole system is cooled down by placing in the ice cold water, and the system is cooled down immediately to get the transparent nanomulsion. By quenching of that temperature, the sudden drop in the temperature induced the turbulence in the system, and the nanodroplet and nanomulsion is formed. So all the 15 formulation, I have conducted some characteristic tests like droplet size, PDI, zeta potential, refractive index, percentage transmittance, and some thermodynamic stability test. The formulation which are red are fail in thermodynamic stability test of centrifugation test and heating cooling cycle test. These all formulation contain higher proportion of oil. That's why they are also appear as turbid, and they are fail in thermodynamic stability. After then I conduct the statistical analysis in which I determine the effect of independent variable on average droplet size. You can see that our p-value is less than 0.05. It shows that all the independent variable have significant impact on average droplet size. The R value is also good. The lack of fit value is non-fit significant, that is desirable for adequate model. And we can see the coefficient value. The oil phase is increasing the total droplet size, while surfactant and glycerol have negative coefficient value. It means they are decreasing the droplet size. Manufacturers also showing the similar result, with increasing the oil, droplet size increasing, while increasing the surfactant and glycerol, droplet size decreases. With response surface plot, we can predict that for minimum droplet size, oil level should be at low with high level of surfactant and glycerol. Then I determine the effect of independent variable on percentage transmittance. We can see that the formulation F2, F4, F6, F12, 13, and 15 are turbid, and they are also unstable because they all contain the higher amount of oil. The statistical analysis of percentage transmittance, we see that the p-value is below the 0.05, so there is significant effect of independent variable on percentage transmittance. Amount of oil decreasing the transparency, while surfactant have good in increasing the transparency of the whole system. Manufacturer shows that surfactant at the certain level increases the transparency of the whole system, while glycerol doesn't have any significant impact on the transparency of the whole system. I also correlate the effect of droplet size and refractive index on percentage transmittance. We can see by increasing the droplet size, transparency of system is decreased. But when we increases the refractive index, transparency increases. Even at higher droplet size, if the refractive index is higher, the transparency is increases. It happens because when we increase the refractive index of the nanomolson system, the reflection of both the phases is minimum. So due to the minimum reflection, the system appears transparent. So by Volkswagen analysis, among all the 15 formulation, F8 formulation was optimum, which have the droplet size 96.5 nm, and it is also thermodynamically stable. With this F8 formulation, I conducted in vitro drug diffusion study and transmission electron microscopy and stability testing. With in vitro drug diffusion study with dialysis bag, I use a marketed formulation. In that, we can clearly see that our formulation have higher in vitro drug diffusion in 0.1 NaCl as compared to the marketed formulation. With transmission electron microscopy, we can see that nanosized droplet are seen, and they are spherical in shape, and they all are very well discrete with each other, and droplet size appear in size range of 8 nm to 120 nm. Then with F8 formulation, I conducted a 6-month stability study. We can see that there is no any significant change after 6 months in the F8 formulation. It shows that our system, our Nanomolson system was stable with almost with 6 months. Then I optimized the independent variable using the desirability function. In that we have set some constant. Amount of oil was maximized, amount of surfactant was minimized, and the amount of glycerol was set as middle value. Average droplet size was set to minimize, and percentage transmittance was set to maximize. Then this with this constant, we calculate the predicted value and desirability value. We can see the desirability value was 0.55, and the predicted value of the independent variables was shown in the table. With this predicted value, I formulated a experimental formulation, and the result of that experimental formulation are in agreement with the predicted value. There is no any significant difference with predicted value and experimental value. This indicates the validity of predicted value of independent variable for the producing the optimized formulation. Finally, with the conclusion, the optimized nanomulsion formulation incorporate higher amount of oil phase, about 52% of oil to surfactant percentage, and Box-Began analysis revealed that all the dependent variable had significant impact on average droplet size and percentage transmittance. Oil phase formed by blending LCT, MCT, and essential oil produce more efficient and stable drug-loaded nanomulsion system, and it can be used as an alternative for commercial MCT oil, which are very costly. And overall, the phase inversion temperature method are simple, cost-effective, and easy to operate for developing the stable and optimized drug-loaded nanomulsion system. And this article is already published in drug development and industrial pharmacy. Finally, I want to thank UGC, Government of India. Thank you very much. Any questions?
Thank you very much. So any question from the audience? No. Manish, I have just a curiosity. Which kind of drugs, if you have an idea or if you have in progress to analyze the loading of other kinds of drugs in this nanoemulsion, and if you have checked the stability of the nanoemulsion to the dilution in the fluids, because the one of the problem and the limitation of nanoemulsion of this colloidal system is the instability in when you do some dilution, so in the body. So have you considered this point?
You are asking about the stability in biological fluids.
Yes.
Am I right? Okay. Generally, the nanomulsion system, the stability of the system can be predicted by diluting the nanomulsion system with in vitro in 0.1 and HCl, which is pH 1.2, which is simulated gastric fluids. So we can conduct the dilution test in vitro. But experimentally in vivo stability study, I didn't conduct.
Okay, so the second question was which kind of drugs do you, in which way do you think to continue this work? So which kind of drugs do you want to load in your system?
Generally, majority of the drugs are hydrophobics. So hydrophobic drugs are the good candidate to load in the nanomulsion system. Apart from this, the food bioactive drugs are very good in loading the nanomulsion system. Generally, the food bioactives are hydrophobics, and they suffer with a high phosphorus metabolism. So nanomulsion system are good for for delivering those kind of candidates, those kind of drugs, vitamins and bioactives and the hydrophobic drugs. Currently, I am working on the delivering the CBD cannabinoids. Cannabinoids, CBD and CBD, yes.
Thank you very much for your answer. We don't have any further questions, so thank you again. And let's move to Mattia Tibboni, the last oral communication. Mattia is from University of Urbino Carlo Bo in Italy. The talk is Nanomedicine Manufacturing, how 3D printing could widen the accessibility to microfluidics.
Hi everyone, good evening. And yeah, I cannot share my screen because Manish is still sharing his.
Manish,
sorry.
Please, okay. Now you could be able.
You should see it now. Okay, so thank you very much, Claudia, for your kind introduction, and thanks to the organizing committee for this opportunity to share with you our results today. And I'm going to talk to you about microfluidic, this innovative technology for the manufacturing of nanomedicine. So the main question is, which is the problem, the main problem during nanomedicine manufacturing? One of the main problems in this process is the scale up. And the scale up usually is difficult to overcome because common bench techniques such as the thin layer evaporation method to produce nanocarriers can produce only limited amount of the formulation. And so the answer to this question, guess what it is microfluidic. I will thanks also Dimitrios that has really well introduced this technology during his keynote talk on the opening of this session. But just a few words about the technology. Microfluidic is an innovative manufacturing approach to produce both nano and microcarrier. He has a precise control of micromixing inside the sub-millimeter channels that can change in form, dimension, and shapes. It has a high production rate compared to classical bench technique. It is easily scalable, and it has been used also for the production of the mRNA-based vaccine against COVID-19. Why it's common to produce 3D printed microfluidic chip compared to the commercially available ones? Because you can have, first of all, a complete personalization by CAD design of the device. So you can choose the dimension, you can choose the mixing ability based on your needs and on the needs of your formulation. You can choose among many different materials that can be applied in the 3D printing process. You can produce them with a lower cost compared to the commercially available one, and the production after a good development is quick and easy. The technology that we use to develop our 3D printed microfluidic chip it's called fused deposition modeling 3D printing. It's a technology that allows to print objects by depositing a thermoplastic material layer over layer. Many different materials are available for this technology, and with modern 3D printer, it is possible to reach very high printing resolution to get also these tiny channels that are needed in microfluidics. So the first project that we performed with 3D printed microfluidic was this one. We printed out a T-shaped microfluidic device made with polylactic acid, and taking advantage of this device, we synthesized liposomes carrying glycyrrhetinic acid, that is a natural extract from licorice. We used soy phosphatidylcholine and cholesterol to as excipient to manufacture liposomes, and we then test them for cytotoxicity, and we test them for drug release and drug permeation in vertical diffusion cells using skin mimicking membranes. And we proved that with the liposomal formulation was possible to have an 8 to 10 time higher permeation of the drug across the skin mimicking membranes compared to a saturated solution of glycyrrhetinic acid. In this work, we have been able, again, to produce this PLA-based microfluidic device that was really cost-effective because to produce one, the cost was less than $1 US. The chip was completely biodegradable because it was made of polylactic acid, and we were able to produce nanocarriers with high production rate. But this chip possessed some limitation due to the material that we used that is easily hydrolyzable with some solvents that can be used in the production of nanocarriers. And so it was not possible to use it with other solvents except few like water and ethanol, and also for only a few times, and then it has to be discharged. So we developed more our idea to have a 3D printed microfluidic chip, and we ended up with this work in which we developed two different passive micromixer that can usually induce wire flow and recirculation, creating a transversal mass transport inside the channels, and so increasing the micromixing and the energy that is needed for the self-assemble of the excipient into the device. And we moved from PLA to polypropylene that was selected as printing materials because it's very chemical resistant to many organic solvents. And thanks to the use of this material with this new chip, it is possible to use also many different solvents such as ethanol, methanol, acetone, acetonitrile, and obviously water. We designed two different chip, one that you can see on the left with the letter A that is relying on a zigzag bas relief, meanwhile the one on the right with the letter B that is relying on asymmetric circular subchannels. Before printing, we did some computational fluid dynamics studies that you can see on the right of this slide to prove the capability of this chip to mix well the solvent in their structure, and we confirmed that the mixing ability was good. So we continued with the printing, and we confirmed by microscope that the dimension were the one that we set up in the CAD design. And in the figure C and D, you can see the chip running with a colored solution inside. Taking advantage of these two systems and to confirm that they were able to produce nanocarriers, we decided to produce two model nanocarriers, one lipid-based using soy phosphatidylcholine and cholesterol, and one polymer-based using PLGA. In both of them, we decided to load as the model drug cannabidiol, thanks to its many pharmacological activities. Here, there are the results of the design of experiment that we applied to evaluate which microfluidic parameters were affecting more the final characteristic of the nanocarriers that we produced. And as we can see, all the microfluidic parameters were affecting the final characteristics, so we were able to fine-tune the final characteristic of the nanocarriers by changing the microfluidic parameters during the production. Here, some other characterization with atomic force microscope that confirmed the size and the shape of the nanocarriers, and some thermal studies to evaluate interaction among the drug and the excipients. So after all, we have been able to develop this personalized 3D printed microfluidic device with a very low cost and with an effective production of model drug-loaded polymeric and lipidic nanocarriers with very high production rate that were up to 30 milliliter per minute. And we strongly believe that with our approach, we're able to increase the accessibility to microfluidic to many researchers, because with this approach, we can have customizable devices with very low cost. And since we believe that in the widening of this technology, we have also left the design for free with in the supplementary materials of our paper. I will thank all our collaborators in the academia and in the industry, and I like to conclude all my talk with this funny image of a teleporting llama, because I think that the 3D printing is the closest technology that we have nowadays to teleporting, because by having a 3D printer that nowadays is very cheap, it is possible to produce the same object all around the world by just sharing the design with other people. And in few hours, it is possible to obtain the same object that has been produced in Italy, also in Australia, in United States, in China, in India, wherever in the world. And thank you very much to all of you for the attention, and I'm open for a question if there are.
Thanks, Mattia. Thank you very much. We have a comment from Vincenzo in the chat. Really nice talk and brilliant graphics. Well done, Mattia. Okay, there is any question? I have just a curiosity, maybe I've missed something of your presentation. When you have shown the last part of your talk, the two chips that you have designed, one is linear, the other one is a circular, okay? Which is the reason of this different design?
So these two designs are designs that both help the micromixing in the device, but in two different ways. So the one that was linear, it was relying on a zigzag pattern inside the channel that helped to induce wire flow in the flow of the solvent inside the chip. Meanwhile, the other one, relying on circular subchannels, it helps the mixing by the collision of the flow where the circular subchannel are in contact at the end of each circle. And so we also evaluate the number of circles that has to be subsequent to have an effective mixing. And we decide, in this case, for 6. But the really good things of 3D printed microfluidic chip is that, for example, for another formulation, 6 circular subchannel are not enough, and maybe you need more mixing. It is possible to increase the number of circular subchannels, but just changing the design and then printing it in a few hours. So again, the personalization is the really strength of the 3D printed microfluidic compared to the commercially available.
So it depends also on the material that you are going to use. So for example, between polymer and lipids, which kind of chip you can choose?
Yeah, it depends on the excipient, and so there is not a strict rule, let's say. For every formulation, you should try, let's say, both of our chip to see which is the one that it can fit the best of your formulation. And for us, for example, the zigzag chip was better for liposomes, meanwhile circular chip was better for polymeric nanoparticles. But we tested only PLGA, so maybe using another polymer, the zigzag would result better. So both of them can be used for both the formulation, and we select these two just a model carrier to prove that our chip were effective to produce nanocarriers.
Okay, thank you. Thank you very much.
Thank you to you.
We have other question? No. Okay, thank you, Mattia. Thank you again.
Thank you.
So now we can move to the last part of the conference, the last short talk, slide and talk. First of all, we have Kriti Shukla. Kriti. Okay.
Yes. Yeah.
So could you share your screen? Kriti is from University of California, Los Angeles. The talk is the efficacy of simvastatin in preventing metastasis of renal cell carcinoma in a chicken chorionic membrane CAM model.
Yeah.
Okay.
Am I good to start? Yeah. Hi, everyone. Thank you so much for having me here. I'm very excited to be speaking with you all today. So my name's Kriti. I'm a chemistry and applied math student from UCLA, and I'm going to be talking today about research that I did in collaboration with our Department of Molecular Pharmacology here also at UCLA, which looks at renal cell carcinoma and different drug delivery options. So for some background into this disease, over 70,000 new cases of renal cell carcinoma are diagnosed each year. Renal cell carcinoma itself actually has a fairly high survival rate, as you can see here, but once it metastasizes and becomes distant metastatic renal cell carcinoma, the survival rate drops to only 13% over 5 years, which is only about 1 in 10 patients. Obviously, this is really deadly in the US because of its high prevalence, but it can be very difficult to develop treatments once the cancer is metastasized, partly because metastatic RCC cells are characterized by their heightened ability to evade immune surveillance and program cell death, which means they can rapidly spread throughout the body and multiply and propagate. And then MRCC tumors are also characterized by their high level of intratumor heterogeneity, which means between tumors, the composition can vary a lot, which makes it very difficult to find uniform treatments that effectively target and eliminate all tumors. But in recent years, there has been more interest in 3-hydroxy-3-methylbutyryl coenzyme A inhibitors, which are also colloquially known as statins, as a potential treatment for various cancers. Statins as a class of drug are more commonly prescribed to lower blood cholesterol. They inhibit the HMG-CoA reductase enzyme, which is part of the cholesterol metabolic pathway. But recent studies have also shown statins reduce cancer-related mortality in patients, particularly in hepatic cancer. There isn't really a lot of research showing their efficacy in renal cancer or metastatic renal cancer, so we performed a high-throughput drug screening to screen for different FDA-approved drugs that may have potential applications in RCC, and that returned 6 different statins, out of which we looked specifically at simvastatin, whose structure is shown here. Simvastatin was especially interesting to us because in the past, in hepatic cancer, it's been shown to induce apoptosis and G0, G1 cell cycle arrest, which makes it a very attractive potential therapy for metastatic renal cell carcinoma. So in this research, we tested the effects of simvastatin and metastasis in a chicken CAM model. The CAM model, it's basically you take these eggs, it can be from any animal, but we look specifically at chickens, and you remove the outer shell layer to expose the chorionic membrane, or the CAM. And on this membrane, you can place your tumor in a matrigel suspension, and the tumors will then circulate throughout the egg and mimic metastasis. And past studies done both in our group and other groups have shown that the CAM model is just as effective as the traditional murine model as an in vivo model for cancer, but this model is much, much faster in propagation. It only has about a 16-day span, so we were able to perform multiple experiments over the course of about 2 months. And in these experiments, we looked at different concentrations of simvastatin administered to chickens that had been given metastatic tumor cells, and we looked at how that affected the growth and spread of metastasis. So we performed immunohistochemistry on the tumors and organs from the different specimens. These slices were placed on a slide. And then here on the top, in this kind of right corner here, you can see that we used flag primary antibody and goat anti-mouse secondary antibody staining, and then DAB and hematoxylin staining. So the DAB gives the slides this brownish color, and that indicates the presence of tumor. And then hematoxylin gives the slides the purplish color in this top row, which kind of shows a nuclear structure. And then the bottom row is H&E staining, which shows, again, structural components at a higher level. And this staining was done by our imaging department here at UCLA. So as you can see in the immunohistochemistry, the control samples with no simvastatin had much more metastasis versus samples with 5 micromolar administered of simvastatin had no metastasis. And these results were replicated in further samples and further studies. So right now, these preliminary indicate that simvastatin is effective at preventing the spread of metastasis for specimens already infected with renal cell carcinoma. These results need to be further confirmed with PCR or flow cytometry just to indicate and confirm the presence of the tumor DNA in the metastatic samples versus the non-metastatic samples, but these results are really promising. For future directions, we are hoping to look not just at simvastatin, but also other statins. Simvastatin is metabolized by the isoenzyme cytochrome P450 3A4, which a lot of drugs are metabolized by, but this is a very effective metabolic enzyme. So the amount of simvastatin disappears very quickly in the body, and there's very little bioavailability. So we're hoping to discover other statins that have a higher amount of bioavailability. A very promising one is maybe fluvastatin, which has an N-heterocycle in its structure that binds very well to many biochemical molecules and thus increases drug delivery efficacy. But that is all for my talk. Thank you guys so much for listening and having me here.
Oh, sorry. Thanks. Thanks to Kriti Shukla, and now is the turn of Valentina Del Genio from University of Napoli Federico II in Italy on demand nanofibers for cancer.
Hi, can you hear me? Okay.
Yes.
One moment. Okay. Can you see my presentation? Okay. Okay. Thank you for. Oh, one moment. Okay. Sorry. Okay. Thank you for presentation. Hi everyone. My PhD project is focused on to produce dynamic nanoplatform for cancer adaptive to response to physiological environment for on-demand drug release. Our self-assembled system is a composite of 3 peptides, B1, B2, and B3. B1 has a role to favoring fiber formation, and it is a structural peptide. B2 was created to enhance internalization. In fact, it consists in a structural sequence, but in this case with a positive charge, and a sequence of peptide for delivery GH625. B3 was created to improve binding with the doxo. In fact, in this case, we have a structural sequence like B2, a sensitive sequence for enzymatic cut, and we add at the C-terminus of this sequence a cysteine to improve binding with the doxorubicin. At N-terminus of each peptide, we add a sequence of 6 alanine and a alkylic tail to improve a hydrophobic driving force for create self-assembling structure. Complex system will be able to release the drug in cancer tissue where there is an overexpression of a proteolytic enzyme, in particular MLP9. Peptides were synthesized and by solid phase protocol using a FMOC strategy and then purified. The covalent bond between cysteine of B3 and melamine moiety of DOXO was analyzed by NMRI spectrum. In particular, in the first picture, we can see a spectrum of reaction between cysteine and DOXO, and respectively spectrum of B3 and DOXO melamine alone. Peptide self-assembly capability of our peptide were analyzed by fluorescence assay used a fluorophore red, and in particular in second picture from panel A to F we can show the CSC graph that show a good results because of the concentration at which the peptide aggregates degrees when the peptide are co-assembled compared when peptides are alone. Each panel showed the thioflavine assay that confirmed the aggregates formation, and in I panel we have a TEM image. In fact, our fiber has a dimension of 100 nanometers. To understand the stability of our system, we performed a CD spectrum of our self-assembled system at different concentration, different pH, and different ionic strength. In the last picture, we reported a preliminary cellular experiment on SKBR cell, and in particular, we compared the fluorescence of DOXO when the DOXO was incubated in the cell alone, and when was incubated in our surface with the bound in our self-assembled system, and we have a good results because after 30 minutes we have a fluorescence of DOXO in a perinuclear zone, and after 1 hour we have a fluorescence in the nuclear zone. So DOXO was released by our self-assembled system. The system would be optimized by other experiments to produce a specific therapy for AIDS patient. So we hope to obtain other good results. Thank you for attention. Sorry, I don't hear. Sorry, don't.
Okay.
Okay.
I had the microphone off. Anyway, thank you very much.
Thank you. Thank you.
One is Maddalena Sguizzato from the University of Ferrara. Topical application of alpha tocopherol containing lipid nanoparticles, skin protection against pollutant. So Maddalena, is yours.
I have just, okay. I think that I'm not able to share because the other presentation is on.
So.
Okay, so now I can.
Okay, now you should be able.
Share. Yes. Can you see my screen?
Yes.
Okay. So good afternoon, everyone, and thank you for the opportunity to share my work with you. And the work is about the topical application of alpha tocopherol containing lipid nanoparticles, skin protection against pollutants. This work was developed in 3 steps: the pre-formulation study, characterization, and the antioxidant, the evaluation on the antioxidant effect on human skin explant. First of all, the solid lipid nanoparticles and so SLN and nanostructured lipid carriers were formulated using different solid and liquid lipid components with the lipid phase of 5 and 10%, tristerine, compritol, presirol, and supposire, were selected as a solid lipid, while mygliol was selected as a liquid lipid. Firstly, the nanoparticles were characterized in terms of size and polydispersity, and the stability of dimension was evaluated in time up to 90 days after production. NLC, after this analysis, appeared as the best stable kind of nanoparticles, thanks to the size stable in time and lower than 100 nanometers. So NLC were selected for the encapsulation of alpha tocopherol. The encapsulation of alpha tocopherol was almost quantitative and was stable in time up to 90 days after production. The nanoparticles were then characterized in terms of morphology and inner structures by cryothem analysis and SAX analysis. Nanoparticles formulated with trasterin, compritol, praesirol appeared as roundish particles, while NLC with supposire appeared most as spherical, resembling to vesicles instead of particles. And this morphology was also confirmed by the SAX analysis to describe the inner structure of the NLC. For the great physical chemical stability of NLC formulated with tristerin, these formulations were selected for the evaluation of the protective effect of tocopherol against cigarette smoke induced skin damage on human skin explants. So the NLC were the skin human explants were treated for 24 hours with NLC and with tocopherol, and after 24 hours the biopsies were exposed to cigarette smoke for 30 minutes. After the exposure, the proteins from biopsies were collected, and the expression of AMYC-oxygenase was evaluated by Western blot. The AMYC-oxygenase is a stress response enzyme under control of antioxidant pathways. So as you can see by the Western blot bands and the quantification on the graph below, the cigarette smoke induce a high expression of AMYC-oxygenase, while in the case of the NLC biopsies pre-treated with NLC and alpha tocopherol, you can see that the expression of AMYC-oxygenase is very low compared to the control biopsies. So with this test, we can confirm that alpha tocopherol is able to protect the skin from the damage induced by pollutants, and in this case of the damage induced from cigarette smoke. So thank you for your attention.
Thanks, Maddalena. So now we have just two short communication. One is from this morning because we had the problem of connection. Arianna Rinaldi, I don't know if she's connected now. No, she's not here. Okay. And then we have Alice Della Valle that unfortunately yesterday was not able to give her short talk. So Alice, you
Good afternoon, everyone.
Hi.
Can you see my presentation?
Just a second. Yes. Okay.
So thank you for giving me this opportunity with my presentation about synthesis and characterization of linear peptides analogs of endomorphin 2 and MET11 caffeine containing Fredinger gamma lactams. Peptides are usually pure drug candidates due to their rapid hydrolytic cleavage, unfavorable pharmacokinetics, and conformational instability. To obtain peptidomimetics more stable of endogenous opioid peptides, the most common approach consists in introducing local or global restrictions in the peptide sequence to form a cyclic scaffold that keeps the pharmacophore functional groups in the right orientation. Fredinger at Merck published in the 80s a series of dipeptides with alpha, beta, and gamma lactam rings, which were widely used to stabilize beta and gamma turn structures. Whose application in biologically active peptide, there have been many. So the aim of this work is the design and synthesis of linear peptides, analogs of endomorphin 2 and MET11 caffeine, containing a Fredinger gamma lactam as a local restriction in second or third position. Fredinger gamma lactam is considered a mimetic of L-prone residue, which is fundamental for the endomorphin 2 for the correct spatial orientation in the Mu receptor binding pocket. So product A1 and A1D are encapsulin analogs with the gamma lactam ring in third position, obtained respectively from L-methionine and D-methionine. Products A2 and A2D are encapsulin analogs with the gamma lactam in second position. And finally, products A3 and A3D are endomorphin 2 analogs with the gamma lactam ring in the second position. As shown in scheme 1, products were synthesized according to a standard solution peptide synthesis. To form the gamma lactam, we use the classical synthesis of Fredinger, which requires an intramolecular alkylation step. The appropriate peptide sequence containing L-methionine or D-methionine was used as a starting material, since the thiomethyl function was transformed into the sulfonium salt and then cyclized to the gamma lactam with sodium hydride. All compounds were tested for the ability of binding and stimulating G-protein coupled mu-opioid receptor. Values of efficacy and potency are reported in table 1, and we can see that the product A2D was the best performer with the Emax. It was able to activate and selectively stimulate the G-protein coupled mu-opioid receptor with an Emax of 143.3% and a potency lower than the reference ligand DAMGO. All compounds were tested also in vivo by tail flick test, and they showed good results. Among them, peptide A2D and A3D were the best performer with a good antinociceptive effect higher than the reference ligand DAPEA. And all peptides were tested also in vivo by formalin test. During the acute and late inflammatory phases, products A3D exhibits a good antinociceptive effect, and if administered subcutaneously, product A3D was the only able to exhibit a good antinociceptive effect during both phases of formalin test. And also molecular docking studies confirm the ability of compound A2D to interact with the Mu receptor binding pocket, assuming a beta turn conformation, while its diastereoisomeric partner A2 was not able to resemble the same behavior in silico. So the only structural discrimination between the two ligands was the chiral center of Fredinger gamma lactam, which is fundamental in the determination of the overall 3D structure at the Mu receptor binding pocket. So in conclusion, we have designed and synthesized a new small library of linear peptides with the gamma lactam of Fredinger in good yields and purity, following a well-established procedure. Data revealed that product A2D was selectivity for Mu receptor binding pocket and had a good antinociceptive effect after intracellular ventricular administration. Molecular studies helped us to understand the key role of Fredinger gamma lactam for product A2D to interact with the receptor, and this could pave the way to more potent analogs of endomorphin 2 and DAMGO. I'd like to thank my research group, my Professor Mollica, Professor Azzurra Stefanucci, Dr. Giuseppe Scioli, and all of you for your kind attention.
Thanks, Alicia. So now we are at the end of this conference. I just ask you 2 minutes for the closing remarks. I'm very happy. And I hope that you have enjoyed this conference. I want just to thank everyone. Alicia, if you can close your sharing. Okay, thank you. I'm going to share my screen. So first of all, I would like to thank all our sponsors, University of Napoli Federico II, Department of Excellence, Chemistry View, Reaxis, Wiley, Elsevier, all the sponsor, Additive and Italian Society for Chemistry to help us to organize this conference and for the great support. I want to thank all the invited speakers for the great talk. And I want to thank to all the attenders, of course. So we reached this year 200 registration, 60 oral communication, 35 posters. And I want to highlight the fundamental rule of Marta de Plan for her support and hosting the conference on the platform, on Zoom platform. I ask to all the organizers that are able to switch on the video. I can see Nunzia Iaccarino, Gabriela. Thanks to Wiley, Chemistry Europe, because all the awards are sponsored by the editorial board of the Chemistry Europe journals. Now is the time of the award. Sorry for the delay. So the poster award for the analytical chemistry is Alessia Lanno from the Department of Environmental Health Science, Magro Negri Institute in Milan. Development and validation of innovative UHPLC-MS method for monitoring the cholesterol synthetic pathway in biological sample. For medicinal chemistry, Amedeo Ruggero. 3D conductive vertical structures for bioelectronic device, and for drug delivery and pharmaceutical technology, Sofia Moroni, direct powder extrusion 3D printing of polydroxybutyrate implant from prolonged drug release. So congratulations to all of you. Now let's move to the oral award, and I want to thank, of course, the scientific committee. The best oral award for analytical chemistry is Federico Fanti from University of Teramo in Italy, screening and quantitative analysis of short chain fatty acid by means SPLCP MS. And we would like also to give a special mention to Mariana Rossetti from University of Rome Tor Vergata. For medicinal chemistry, the best oral award is for Francesco Fiorentino from University of Roma Sapienza. His talk was Drug Discovery Meets Native Mass Spectrometry, and a special mention is for Pier Michele Cubari from University of Groningen, Netherlands. Finally, for the session of today, for pharmaceutical technology and drug delivery, the best oral award is from Vincenzo Taresco, University of Nottingham, who talked about polyglycerol adipate. And a special mention is for Sofia Prarasia from University of Padua, who talked about the brain delivery of an inhibitor of the potassium channel Kv1.3. So thank you again, all of you. I want to thank you again, all of you. I hope that we can see all of you in Naples next year for the iTunes meeting for young chemists in biomedical field that will be in 2022. So it will be the 3rd edition. So thank you, everyone, and goodbye from all the organizers.
Goodbye, everyone, and thank you.
Goodbye, everyone.
Goodbye, everyone, and thank you for being with us.
Thank you very much for the opportunity.